Okay, everyone, we will wait just 1 second to get started. We'll wait one more minute, let a few more people get online, and then, yeah, we'll go ahead and kick off the webinar. So this webinar is part of a series between us, the 3Rs Collaborative, as well as the IQ Consortium. This is part of our 2026 MPS webinar and workshop series. And for the next 2 days, we'll actually be revisiting liver MPS. So as a disclaimer, this webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussions of competitively sensitive subjects, such as costs, prices, sales, product marketing, and other confidential information. In addition, neither 3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales during collaboration activities. So now that that disclaimer is out of the way, I'd love to give just a brief introduction first to us and this webinar series, and then I'll hand it over to a representative from the IQ. So as the 3Rs Collaborative, our broader group really focuses on all 3Rs: refinement, reduction, and replacement. But this initiative is really focused on MPS and collaborating to accelerate the adoption of MPS in scientific research. Yeah, and we also seek to increase regulatory use of MPS where scientifically justified. It is a very large group. It's actually the largest group within the 3Rs Collaborative. These are pictures and or logos of all of our different member institutions. We have over 50 of them. They're primarily commercial developers, but also include consultants, enabling providers, regulators, end users, and more. We've got 4 key aims. One is to provide thought leadership and expert consensus. Second is to facilitate discussion, collaboration, and sharing. Third is to create engagement between developers and regulators. Fourth is to develop external partnerships and collaborations. And fifth is to create resources to facilitate increased engagement with commercial MPS. We accomplish these goals via 3 key work streams. One is our regulatory group, second is our technology expo, and third is end user engagement. And I'm going to go through the 4 key impacts that these groups have made over the last 6 years. I think we launched at the end of 2020. So first of all, I want to point out our website. The majority of our resources are easily and quickly found on our website, 3rc.org/MPS. This includes everything from an overview to discussions about various acts, presentations, our tech hub and publication hub, which I'll go over, how to join, and more. So this is a really good resource. This is also where you can find recordings of all of our past webinars. You can do this either through the MPS Hub or through our event tab. So if you want to go back, see how much liver MPS have changed, you can see those very first webinar and workshops from 6 years ago. The second big impact that we're doing is about advancing use of MPS in regulatory submissions, especially to the FDA Center for Drug Evaluation and Research. We've done a lot in this area. We have a publication outlining the use of MPS in 3Rs and regulatory applications with perspectives from developers and stakeholder responsibilities. We have quite a large DILI drug-induced liver injury project, which Nikisa Sandria will actually outline today. We're working with 6 core stakeholders, 8 developers. You'll learn more about that. It's very exciting. We do a lot of thought leadership. We lead discussions, presentations, panels at a variety of conferences, and we even have a benchmarking survey about using MPS in discovery and development. Third, we're actually engaging with end users and regulators in this workshop series. This kind of showcases all the different topics we've done in the past. Today and tomorrow's Liver MPS, and then save the dates for September 8th and 9th and December 1st and 2nd for future topics. Our fourth and final big impact is about our tech and publication hub. This is where you can find all of the developers after this webinar. You can filter by tissue, disease, or species. And we also have a publication hub that has over 400 MPS publications from those developers, again, with really useful filters for you to engage with. During this webinar, if you can use the Q&A to submit questions, that's the best way to do it. If you have to use the chat, we'll also monitor that as well. Visit our website, email our program manager, Eliza. I'm Megan LaFollette, I'm executive director. I'm stepping in. Eliza has a little cold today, but email Eliza if you need anything. You can also email me if you're interested in joining or becoming one of our sponsors. And with that, I'll turn it over to the IQ representative.
My name's David Kukla. I'm part of the IQ MPS affiliate, so I just kind of wanted to take a few minutes just to give a little overview of the IQ. So the IQ is a collaboration of pharma and biotech companies created as an affiliate within the International Consortium for Innovation and Quality in Pharmaceutical Development. And the IQ has four main aims. One is to develop external partnerships and collaborations to help enhance the inclusion of industry priorities. Two is to serve as a thought leader for MPS developers and stakeholder organizations. Three, to provide a venue for appropriate cross-pharma collaboration and data sharing. And then finally, to create focused engagement between industry and regulatory agencies on the current status and the evolving field of MPS in industry. So the IQ is currently composed of 28 member companies with over 150 active participants, which represent experts from drug safety, 3Rs, ADME, as well as PKPD. So here's just a quick overview of our 2026 Executive Committee. Rhiannon David was our previous chair in 2025. And then Kim Homan is our current chair with Philip Hewitt as the vice chair and James Gossett as the vice chair-elect. So here's just kind of a breakdown of how we structure the IQ MPS. Basically, the executive committee provides leadership and guidance in the management of IQ MPS business and affairs and implements strategic plans recommended by the steering committee. And then the steering committee is made up of two representatives from each one of our member companies, and they are the primary decision-making body of the IQ. And then within the IQ, we have 5 major work streams, which I'll quickly highlight on the next few slides. And then within each one of these work streams, we have several sub-teams which address specific critical issues. So the first work stream is regulatory engagement, and their mission is to build regulatory confidence in MPS and align on a common understanding of how MPS data should be handled in regulatory submissions, basically by sharing industry knowledge of MPS utilization with regulators and collaborating to overcome challenges utilizing MPS for drug development applications. The second work stream focuses on publications, and their mission is to outline the contemporary pharmaceutical industry perspective and considerations for developing, evaluating, and characterizing MPS models to support drug discovery and development. Then the pilot project workstream works to initiate near-term proof of concept studies for data sharing or prospective collaboration for qualifying MPS systems, basically based on kind of a specific context of use. And then also the strategic partnerships and communications workstream aims to develop external partnerships and collaborations to help enhance inclusion of industry priorities by trying to make the IQ MPS the partner of choice for pharmaceutical MPS education, alignment, and engagement. So if you have any questions, please feel free to contact us at the email provided on the slide, or you can keep up to date with the IQ MPS's initiatives on either our website or our LinkedIn page. Thank you.
Thanks, David. So I'll be talking about actually, we have in CDER FDA. I'm from CDER FDA in the Office of New Drugs, and we have a public-private partnership. I'm sorry, the phone is. We have a public-private partnership with both the 3 Rs Collaborative as well as the IQ MPS. So it's good that this webinar kind of is a joint thing between both of them. But I'll be really focusing on the project that we have with the 3 Rs Collaborative under the PPP, and really it's based on trying to gain confidence in microphysiological systems. So why were we interested in this project? Well, we did an analysis of the landscape of NAMs submitted to CDER in drug applications, INDs and NDAs and BLAs, and what we found was that when we searched for keywords which are in this table here, that describe in vitro, in silico, in chemical, and other, and that's kind of like what we are using for our definition of NAMs, right now. And we used some keywords that would potentially be present in the study title. What we found is that there are, predominantly in vitro and in silico studies that are submitted to FDA, and very little of the other ones. So really, these are the two categories. And in silico really represents, it's not really representative because for the most part, these are, tests that are used for qualifying impurities and genotoxicants that might be there as degradants or impurities. And so, this number is sort of artificially inflated. But when we're looking at the number of in vitro NAMs, and that includes all in vitro NAMs, the spheroids and organoids and microfluidic systems and everything, it's about 1,200, and that was for a 15-year period. And we're doing a sort of like updated analysis for the sort of like since 2024, the end of 2024, so last year. And while you think that that's a lot, it's actually not a lot. If you look at the bottom here, it represents 0.25% of the total number of studies that are sent to FDA. So really, we don't have not many NAM studies, even if they are very general, kind of like we're lumping in, stem cell sandwich cultures, co-cultures and everything. Very few are submitted to FDA. So we need confidence. And so what we decided to do was to start this public-private partnership with the 3 Rs Collaborative in order to evaluate commercial NAMs, because we thought that if people were to submit things to FDA, it's likely that they would be using commercial NAMs, and then also we wouldn't have access to sort of proprietary NAMs to evaluate them. So we really focused on these commercial NAMs, and so the objective of this project was to build confidence by doing a cross-platform evaluation of liver MPS models. And why did we choose the liver? Because, there had been surveys done, publications and everything kind of listing that liver was the most advanced model. That the liver was the most advanced model, and in a way, if you think about it compared to the kidney, the brain, the liver is more simple. And so, if we can understand how get a sense of the liver sort of, and be able to mimic the liver, then it would be conceivable that other organ systems would be doable. If we can't do the liver, then that's going to be a problem. So for this project, we decided to sort of use some harmonized principles and design a common protocol, while really accepting that each platform that was going to be part of this project was going to be different. So really the variety was embraced in this project, because we hoped that people would, the participants would use their platform the way that they see fit, but that they would try to adopt or adapt their model to, stick to the common protocol that was developed. And so we had to kind of use a context of use, which is really the sort of principle that a lot of these NAMs are based on for their value in regulatory, decision making. And so we decided that the way that we were going to, the question that was going to be answered was whether, this liver microphysiological system could serve as a retrospective analytical tool to study drug-induced liver injury that's observed in clinical trials. So you observe something in clinical trials, you go back to the MPS and say, instead of doing an animal study, now we're going to do a microphysiological system assay in vitro model. Can we understand what's the mechanism of this toxicity, and can we somehow, keep moving forward with the drug development plan? And so there were a number of deliverables for this project, and one was to submit an iStand qualification submission, and that was something FDA wasn't part of, but that was sort of like a side project that as the data that was generated could be used to submit a qualification package to FDA, and it was submitted, and this past January it was the letter of intent was accepted. Papers are being published. So right now a paper has been submitted on the process that was used to kind of develop this project, and once the results are finalized, the analysis is completed, the results will also be published. And what we're hoping is in the future that we will learn something about best practices to support guideline development, at least for the area of liver microphysiological systems. And so there are a number of stakeholders that are involved in this project, which makes it very, good for us at the FDA, because really what happens is that we are working with a nonprofit organization, the TheWell Collaborative, but there are end users such as pharma and consultants that are involved. The Critical Path Institute is involved. NIHS is also involved, and then of course the developers. And so looking at the timeline for the progress, as I said, we started in 2023 with the PPP, and then shortly after the Critical Path Institute sort of joined in to start helping with the protocol development, and really they kind of moved the sort of letter of intent and the qualification package forward, and they helped with the development of the protocol, and then NIHS joined in, which really was critical because they have a lot of the scientific expertise, and they helped in the sort of development of the list of drugs, which I'll mention in a minute, and they are going to be helping in the independent analysis of the study results. And so the studies were started to be conducted sort of like at the end of sort of last year, basically around this time. And the letter of, as I mentioned, the iStand submission was submitted and accepted. And right now we're kind of working on the analysis of the data. So 8 commercially available liver platform developers were involved in this project. They're listed here, and they represent a broad range of technical and biological complexity. Some are plate-based and some chip-based systems. There's static and flow-based configurations. Some use primary hepatocytes or induced pluripotent stem cell-derived hepatocytes or co-cultures with non-parenchymal cells. So really, there's a variety of platforms, which again, they have the liver in common, but that we wanted to look at the variety, the variability, because what we're interested in is what is the variability that we might expect. We are not comparing one platform to another. We're trying to evaluate how these platforms sort of measure the same endpoints, basically. So the drugs that we selected, there were 8 drugs. They could fall into like 4 pairs of drugs, and they have different indications. They're from different categories of drugs, and in each pair, one was considered to be the more toxic one, which causes drug-induced liver injury. The other one is kind of like the control, which is less drug-induced liver injury. However, these drugs were blinded and sent to the participants of the study, the 8 platform developers, as a set of 8, not as pairs. So they just received 8 blinded drugs, so they didn't know which drugs they were evaluating. And so the study was kind of harmonized in the sense that the solvent was maintained at a certain level, the MSO, and so if they sort of went to the sort of maximum Cmax that we had proposed, which was 10 times the Cmax. The number of concentrations per compound, however, varied. Some people used 4, some people went up to 9. The repeated dosing was between 1 to 14 days, depending on the platform and their capabilities. Number of replicates varied, from 3 to 8. They also had different standards for documentation, which, and data collection, which really we tried to address sort of later on for our independent analysis, which is in the last bullet point, which is where we're at right now. And that's going to be conducted at NIH, at NICETM, basically. So the endpoints that were evaluated were based on what their platform was able to evaluate. So here you can see there are a number of different, ATP, albumin, urea, LDH, ALD, AST, typical sort of like measurements. But not all companies measured everything. Some could measure up to 7 endpoints, whereas others could measure just one endpoint. And so, but that was fine because again, we were looking for the really, what each platform could do, because it's likely that each platform might have, different strengths and weaknesses, but they might be able, and depending on the context of use, again, go back to that concept of the context of use, that's what really matters. So there were a number of different endpoints that were evaluated based on the platform. And, the goal was to really mimic what FDA might see. And so we asked initially that each developer submit a study report to NIHs with their conclusions, evaluating all the 8 drugs together, not really the pairs of drugs. So this is where we're kind of at right now. We're at the stage where we want to get now the sort of data, sort of the raw data from people. So the preliminary observations that I'll mention, right now are really based on these self-reported, results. And we really didn't tell anybody, didn't give them any guidance as to how to interpret their results or report it. We wanted them to be, using their own judgment as to how they would interpret the results and report them initially, so that then that would allow us to determine how we might want to standardize the format so that it would be applicable to everyone. So again, right now the data that's being reported is really based on self-reporting and lack of knowledge of the what the drugs were, even the pairs of drugs. So what we kind of observed is that the reporting was obviously going to be also variable. Some people said that a drug had caused high drug-induced liver injury or maximal drug-induced, or some had even a medium, moderate, low. And some even said it was inconclusive. So really the terminology was not standardized. And so, which is fine. We knew that in a study submission that was sent to FDA, people were going to be reporting things like this. And so that was the whole goal of this, was to see, okay, if they report things like this, what does that mean, so that we can then try to standardize it in a way that we can at least try to understand what the results mean in the context of a particular submission. Some companies recommended that further testing with additional donors, concentrations, or assays, would be necessary. And really, there were some changes that were made as well, to the protocol by some people when they felt that it was necessary. So, we sort of wanted to allow for that level of freedom, at least at this stage of the sort of testing. And again, this goes back to the fact that variety was considered important and really something that we needed to work around, because we're not trying to standardize anything. We're just trying to understand variability. So what are some preliminary results? I'm not going to show the data, but I'm going to give you the summary of what these preliminary results say. So the commercial technologies vary greatly in study protocols, assays, and reporting, even when evaluating the same context of use. Most platforms and their internal analysis successfully distinguish the safer compound from each analog pair, although the compound ranking was less accurate across companies when considering all the 8 drugs together. There was a trend showing that the more endpoints that somebody evaluated, so, as I said, some people had up to 7 endpoints, some people had 1. The more endpoints that were evaluated, the more accurate was the DILI prediction, both in terms of the DILI call as well as the ranking accuracy. So ranking between the low and the high, oops, that was a 6-7, low and the high, DILI for that pair. The data presented are preliminary. So, right now, what I just mentioned is really based on our initial observation of the self-reported studies, and we are going to evaluate this further and actually in more detail. So an independent analysis of the raw data is going to be conducted. An analysis of potency and liver injury and severity is going to be conducted. An analysis of variation within and across platforms will be conducted. liver enzyme leakage biomarkers potentially are going to be looked at, and hepatocellular function indicators, because some transcriptomic analysis will also be conducted, since we asked for samples to be sent to NIHS. Now, the limitations are, this is initial study reports. They did not group the drugs as structural analogs. Interpretation across multiple assays to conclude whether a drug causes DILI or not is going to be different for each company at this stage. Some companies tested at higher concentration to 10 times Cmax, which may have impacted their conclusions, because also they exceeded the DMSO limits that we had established. Companies were able to customize components for the assay, which may be adding to the variability, but again, that was to be expected, and it's not a problem. We just need to figure out how to work around that. We obviously did not use a huge number of compounds, but we had to kind of do something that was sort of a starting point, because this is really a proof of concept here that we're doing. So, the limited number of compounds might be considered as a limitation. And there are obviously technology variations which may not enable direct head-to-head comparison between, platforms, but that was not really the intent here. But we also, the experiments were run by expert developers, so we didn't take into consideration that if these are commercial platforms, transferred to other laboratories which would purchase them, what their variability would be. That would be something that would have to be also taken into consideration in the future if somebody buys the platform and then uses it for regulatory submission to demonstrate. And this is why sort of setting the minimal kind of like standards to evaluate the performance of the methodology is important. But I think that despite these constraints, the study is positioned as a proof of concept for regulatory qualification pathways, as well as for how people can submit their studies to FDA. Because again, qualification, I just want to highlight, qualification is not a requirement, neither is formal validation, but a kind of following some criteria for validation is important, which is something that I didn't talk about here, but in a guidance that we recently published. And we want to hopefully develop some best practices. So the work really demonstrates how cross-platform blinded and context of use-driven evidence generation can really build confidence in liver MPS, or help to build confidence. We haven't gotten there yet, but this has been a very good learning sort of exercise for both the participants in the project as well as for regulators. And again, it provides a practical blueprint for how MPS and NAMs can be evaluated, and it supports the 3 Rs and strengthened human relevance human relevance and safety assessment. And I'd like to acknowledge the, in CDER, the Farm Talks staff, as well as the CDER NAMS Coordinating Committee, NIHS, 3 Rs Collaboratives, Critical Path, the participants, and IQ-MPS here, because also they helped in the keywords identification that we did for our work. And with that, I will end. I think I may have run a few minutes over, but thank you very much.
Okay, hello everyone. It's great to be here today. As Alisa said, my name is Nick Nicolaou. I'm the head of cell biology here at pixlbio. And what I'm going to present over the next 15 minutes is our recent work on the development of functional iPSC-derived liver models for drug discovery and safety studies. But first, a few words about pixlbio and who we are, as we are relatively new. pixlbio is the strategic merger of 2 biotech companies that have been in the field for quite some time now. In the UK, we had Definigen, which now forms our stem cell biology unit here at pixlbio, and consists of cell and molecular biologists who develop all the in vitro models that we offer now as a company, including hepatocytes, hepatic stellate cells, macrophages, intestinal organoids, etc., etc., etc. In Sweden, we had Phenaros, which now forms our large-scale phenomics and AI unit. And the team in Uppsala is responsible for the generation of billions of morphological data points to accurately predict cell state, toxicity, and mechanism of action using both our iPSC models that we generate in the UK, as well as using a plethora of immortalized cell lines across all tissues that we have in-house. And these 2 units now operate as one big family, creating a unified platform, as we say, for predictive and scalable drug discovery studies. The focus for this presentation today, as expected, is the liver models that we have generated in-house. So I will briefly describe the rationale behind the setup of our company. And this rationale is actually based on the challenge that we have all been facing in the pharma sector. And this is the need to identify relevant in vitro liver models for drug discovery and safety studies. And that need is mainly due to the limitations that we have been all facing with the current models that we use. And actually, these limitations is the reason that forced scientists towards the development of iPSC-derived models almost 20 years ago, including iPSC-derived hepatocytes. However, and despite their promise, those iPSC-derived hepatocytes initially failed to recapitulate human hepatocyte function, as many of you know. And that was mainly due to their immature phenotype, which was associated with differences in drug metabolism, transporter, and toxin response pathways compared to primary human hepatocytes. And this is where pixlbio comes, because what we have done over the last 4 years in particular is to significantly optimize our three-step direct differentiation platform. And now we are very happy and proud to offer mature iPSC-derived hepatocytes with enhanced functionality, consistency, and scalability properties. And with this in mind, I will move into the data that we have presented here today for you. And for us, the first approach was to actually compare ourselves, our optimized iPSC-derived hepatocytes to the conventional iPSC-derived hepatocytes that are publicly available. And for this reason, we performed RNA sequencing analysis. We compared our cells, our pixhep, as I will be calling them from now on, to protocols that are publicly available. And we generated iPSC-derived hepatocytes followed by RNA sequencing analysis. And the results are presented in this slide here. In the first figure, you can see how our cells, our optimized pixhep, cluster way closely together compared to the traditional iPSC-derived hepatocytes. And this is indicative of the really low batch-to-batch variation that we are aiming as a company. And on top of that, you can see that these two groups cluster way differently, indicating significant differences at a whole transcriptome level. And indeed, when we performed additional pathway enrichment analysis, we identified a variety of pathways that were differentially regulated between the two groups, notably those impacting drug metabolism, cytochrome 450, type 1 diabetes, so metabolic pathways related to the liver, metabolism of xenobiotics, and bile secretion. And these were the data that forced us to actually try to investigate how our cells compare to the cell types that almost everyone is using in the pharma sector. And these models are either hepatocellular carcinoma cell lines, as HepG2s, or, of course, as you all know, primary human hepatocytes, the gold standard in liver biology. The first approach for us was to investigate the expression of the gold standard hepatocyte maturity markers, including albumin, hepatocyte nuclear factor 4 alpha, and alpha-1 antitrypsin. And as you see, in all cases, our cells, our pixhep, express comparable levels of these markers to what we see in 2D monolayer primary human hepatocyte cultures. But what I would like to focus on is this marker here, alpha-fetoprotein. You probably all know that AFP, alpha-fetoprotein, is a fetal hepatocyte marker. It is only expressed in fetal hepatocytes. And what this data set here shows today is that our cells have escaped that fetal hepatocyte stage that iPSC-derived hepatocytes were demonstrating in the past. And now our cells are more mature and actually comparable to primary human hepatocytes, the mature version of hepatocytes, of course. On top of that, our cells secrete albumin to a high level. Quite frankly, when our cells are kept in a 2D monolayer culture for 12 days, they start secreting more albumin compared to what we see in primary human hepatocyte cultures. Again, this is evident of the rapid dedifferentiation that those hepatocytes undergo when they are put in a 2D monolayer platform. And what's more, our cells can be kept into this setting for even more prolonged periods of time. And they secrete more and more albumin. And that highlights the enhanced functionality and consistency when it comes to these cultures. And here we have data for up to 24 days post-thawing of the hepatocyte vials, but I will visit this topic a bit later in this slide deck. The second metabolic pathway that we were focusing on as liver biologists was the urea cycle pathway. a liver-specific pathway. It is requiring the parallel activity of all these 5 enzymes here that are highlighted in green. And our cells are expressing all these 5 urea cycle enzymes to significant levels. These levels are again higher to what we see in HepG2 cells, and they are very nicely reflected at urea secretion level. So when we check urea secretion into our system in our PixHep and we compare that to HepG2 cells and primary human hepatocytes, we found that our cells were secreting way higher levels of urea compared to the cancer cell lines and more closely related to what we're seeing in primary human hepatocytes. And in a very similar pattern to what we observed with albumin secretion in the previous slide, urea secretion was going up over time into our system and was remaining stable after 24 days in culture. Quite importantly, our cells demonstrate a fully functional phase 1 and phase 2 metabolic gene expression profile that is comparable to primary human hepatocytes. We have a plethora of data ranging from RNA sequencing data up to functional activity. For the interest of time, I have put just the mRNA data that we have generated following qPCR and RNA sequencing analysis. But what you can see in this slide is that our cells express all the phase 1 metabolic genes as well as all the phase 2 metabolic genes, and these levels are actually comparable to what we see in primary human hepatocytes. In figure A, you see that our cells have really high levels of CYP3A4, CYP2B6, CYP2C9. This is comparable to what we see in primary human hepatocytes, way higher to what we observe in HepG2 cells. And the same pattern exists at phase 2 metabolism as well. Our cells express all the enzymes involved in the UGT family, in the GST family, in the sulfosyltransferase family, and these levels, again, are comparable to what we see in primary human hepatocyte cultures. So we know that our cells are fully functional. They are superior to HepG2 cells, to cancer cell lines, and very primary-like. But what makes them unique is that they come with additional benefits and advantages compared to primary human hepatocytes, again, when these are put in a 2D monolayer. And the first benefit lies on the expansion capacity. This is why we're using iPSCs, because we can generate millions, if not billions, of cells coming from the same donor. So we never run out of hepatocytes in this regard, which is something that you cannot achieve with primary human hepatocytes. And when this benefit is combined with donor diversity, where we can incorporate additional iPSC donors, then we can track donor diversity in a very controlled way in a 2D setting. As pixlbio, we have a variety of wild-type donors, iPSC wild-type donors that we have successfully differentiated towards hepatocytes. Here we are presenting 3 of them. We have named them PixHep1, PixHep2, PixHep3. They are of Caucasian background, both male and female donors in this regard. And as you see in this figure, all 3 of them express really high levels of albumin, high levels of alpha-1 antitrypsin. They also secrete high levels of albumin. But when it comes to their phase 1 metabolic gene expression profile, this is where things start to be quite different. And these differences at the gene expression profiles of the phase 1 genes is what we are really interested in as scientists, because we are interested in the donor diversity in order to identify how different drugs and different modalities work in different populations. The second benefit is, and I have touched upon that already, is the prolonged cell culture stability that our cells demonstrate even at a 2D monolayer. I have shown you data already where our cells remain stable for up to 24 days. We have further stressed that setting, and we have kept the cells for up to 34 days upon thawing of those cryopreserved vials. And here you see that the cells remain stable and functional over those 35 days. They keep secreting, expressing really high levels of albumin, really high levels of HNF4 alpha, and they keep secreting high levels of urea, which is quite important when you want to investigate or characterize drugs of slow clearance or drugs that are not that potent as you should expect. And when those cells are combined with our phenomics platform, then things become quite impressive, and we're very happy about that. And in order to demonstrate the functionality of this combined platform, what I'm going to present over the next few slides is a case study that we performed in collaboration with AstraZeneca. This study applied our wild-type PixHep, and we treated our cells with 20 to 25 drugs of known drug-induced liver injury liability. Those drugs, as you can see, were ranged between from high, most DILI concern, to medium, less DILI concern, or no DILI concern. Treatments were taken for 48 hours in a dose-dependent manner, and we performed a variety of both orthogonal assays as well as cell painting and phenomics at scale. And let's start with the orthogonal assays, which were somehow predictive. We run simple cell viability studies measuring ATP content and albumin secretion. And here I'm presenting some of these drugs that we evaluated using our wild-type PixHep. You see 7 of these drugs. In shades of purple, we have the most DILI concern, the high DILI concern drugs. In shades of pink, we have the medium DILI concern. And in shades of gray, we have the no DILI concern. And those cell viability assays were predictive to some extent because they were able to distinguish between the high and the medium DILI concern and the no DILI concern drugs. So we were actually able to measure differences at ATP content. And that was very nicely reflected at albumin secretion level. We were seeing that the high DILI and the medium DILI concern drugs, the cells were secreting less albumin following treatment with these drugs compared to vehicle or the non-toxic ones. But it was not entirely predictive. That was the problem here. And that was because there were still drugs in that family of drugs that we tested of either high DILI or medium DILI concern. And those simple cell viability assays failed to confirm. And now we know that that problem was not because of the cells, but rather due to the sensitivity of these assays. And this is where phenomics comes. Because when we perform phenomics using the same setting again, the same drugs, same concentrations, same time points, and same cells, of course, results were significantly different. Here are some examples from clozapine and olanzapine. Clozapine is a high DILI concern drug. Olanzapine is a medium DILI concern drug. They failed to demonstrate or confirm DILI in vitro using ATP content as an endpoint assay. However, when we perform cell painting, we are able to clearly separate these drugs in the morphological space. Here is all the cells that have been treated with DMSO, our control cells, and here you see how the olanzapine and clozapine treatments have separated into this MSD graph. And a very similar pattern was observed with more drugs. So in the case of cyclosporine, chlorpromazine, a high DILI concern and a medium DILI concern drug respectively, again, they were failing to demonstrate a clear effect when it was coming to cell viability, a very mild effect in the case of cyclosporine in particular, just 15%, as you see from this graph. But again, cell painting was able to distinguish the effect between DMSO and the treatments, and that was very nicely reflected at those morphological changes captured in this graph here. And the most important bit in this regard is that all this data by cell painting capture morphological changes at sublethal doses, so we don't have to go really high with the concentrations of the cell of the drugs. And to finally evaluate whether that sensitivity was due to the cell painting, due to the cells, or due to the combination of both, we generated a dropout model where a given treatment is dropped out, and then we were trying to bring it back and predict if it belongs to the right treatment, the right drug. And this model consisted of the PixHep data that we have generated in-house, alongside primary human hepatocyte data from publicly available studies. And again, we use the same time points, 48 hours of treatments, the same drugs, as you can see in this graph. The only difference was between primary human hepatocytes and PixHep. What I would like you to keep from this graph is that the longer bar, the better. And this is the data that we generated following the primary human hepatocyte analysis. But when PixHep comes into play, we result in a model that is 20% better in predicting toxicity compared to primary human hepatocytes. So to summarize, I hope that I have shown you that our cells are superior to conventional lipase-derived hepatocytes and hepatocellular carcinoma cell lines. They are very primary-like, but they offer unique benefits in large-scale screening, including donor-to-donor diversity, long-term drug effects. And when these cells are combined with large-scale phenomics, we can capture subtle signals that are missed by just cell viability assays. And that enables overall to generate predictive models that are better compared to what we have at the moment. I would like to thank you very much for your attention. I don't think we have any time for questions, but happy to go through this during the roundtable. And again, thanks for being here.
Thanks so much. Hello, everyone. So thank you for giving us the opportunity to present one of our commercially available liver MPS during this webinar. So I will start by briefly present our company. So PredictCan was founded in 2021 in Montpellier, in France. And our mission is to improve drug safety assessment through personalized human models. And today I'll present our commercially available DILI platform, which captures inter-individual variability, contextualizes findings through benchmarking against reference drugs, and help uncover mechanism of liver toxicity. So at PredictCan, our goal is simple. Make drug safety assessment more predictive for real patients. Our patented technology combines multicellular liver spheroids with donor-specific human serum to create personalized liver models that capture human diversity. So rather than measuring an average response, we reveal how different individuals respond to the same drug. This enables us to identify susceptible subpopulations, contextualize findings through benchmarking against reference compounds, and uncover a mechanism deriving DILI. The result is a more human relevant and actionable approach to DILI assessment. So, as you may know, a central challenge of DILI is that patients do not respond to drug uniformly. Differences in metabolism can strongly influence liver toxicity risk. So our platform tackles this challenge by conditioning liver spheroids with donor-specific human serum, which derives HepG2 cells toward a more functional hepatocyte-like phenotype. So as shown here in the left side, we observed enhanced basal expression, basal CYP expression, and a metabolic profile characteristic of active liver cells. And most importantly, the donor-specific component allows us to capture real inter-individual variability. So here in the right, you can see inter-individual variability. So you can see that the same compound induce CYP activity differently across donors. And this transforms the model from a standard toxicity assay into a platform capable of revealing patient-specific responses and identifying populations at greater risk of DILI. So, as we know, for long-term liver safety studies, model stability is essential. So our serum conditioned liver spheroid rapidly form a compact organized structure within 3 days, and then remain remarkably stable for nearly 2 weeks without uncontrolled growth or expansion. So this gives us a robust and reproducible platform for repeated dose studies, ensuring that the response we measure reflects drug effects rather than changes in the model itself. Stability is a prerequisite for predictivity, and our platform delivers both. So next, as we know, for a DILI platform to be useful, it must be reproducible, not just once, but across operators and experimenters. To test this, independent studies were conducted by different experimenters on different days using the same compounds, serum, and cell batches. Across all 3 reference drugs, we obtain highly consistent results. as you can see, and this demonstrates that our platform delivers robust and reproducible readouts, providing the reliability needed to support confident drugs safety decisions. So many in vitro models claim physiological relevance. The real question is, can they predict patients' outcome? And to address this, we prospectively tested our platform using serum samples from breast cancer patients treated with ribociclib. Personalized liver spheroids were generated before treatment and then challenged in vitro. And here you can see the result was striking. Actually, the responses observed in our model reflected the clinical outcomes and correctly predicted DILI susceptibility. So this study moves beyond mechanistic modeling and into patient prediction. So it provides compelling evidence that our platform can bridge the gap between preclinical testing and clinical reality, opening new opportunities for personalized drug safety assessment. So the key strength of our platform is that it moves beyond average biology. So instead of generating a single toxicity value, we capture a spectrum of responses that exist across different individuals. So here, as you can see, at clinically relevant exposure, donor responds very differently to the same compound, closely mirroring what is observed in the clinic. So this data allows us to stratify susceptibility, predict toxicity severity, and build individualized responses profiles. So in other words, we can identify not only whether a drug may cause liver injury, but also which individuals are most likely to be affected. And this capability has the potential to shift DILI assessment from population average toward precision toxicology, enabling earlier identification of at-risk populations and more informed drug development decisions. But identifying at-risk individuals is only part of the challenge. The key question is what derives their susceptibility. And to address this, we build personalized multicellular models from tox-positive patient serum, including liver spheroids and immune cell types such as macrophages and dendritic cells. These are directly compared to the tox-negative individuals under identical conditions. And through combined transcriptomic and functional profiling, we can dissect the pathway and align differential drug responses. And this approach enables a shift from descriptive toxicity to mechanistic understanding at the patient level, providing actionable insight into the biological basis of DILI. And finally, we can benchmark your compound against well-characterized reference drugs within a clinically relevant DILI framework. So this allows us to position a drug on a clear scale, risk scale, low, moderate, or high, based on how it behaves across a diverse donor population and how it compares to known compounds. So the result is structured reference-based assessment that support confident go, no-go decision in drug development. So now let's say that you are in early development and you don't know the Cmax of your drug, and this is actually a common challenge in early development, meaning in the absence of clinical exposure data, which limit conventional safety assessment approach. Here, our platform addresses this by benchmarking compounds against a reference DILI risk map and independent of precise clinical pharmacokinetic. This enables us to assess whether efficacy-relevant concentrations may already overlap with regions of potential liver toxicity risk. In doing so, we provide early biologically grounded insight into safety margins and support more confident decisions making at a stage where traditional framework cannot yet be applied. So in conclusion, our serum conditioned spheroid model delivers a complete layered framework for DILI assessment. It starts with early clinically relevant risk classification to support decision-making in development. It then extends to population-level susceptibility, capturing inter-individual variability to identify not only whether a drug is risky, but for whom it is risky. When a signal is detected, we can move into mechanistic resolution using multi-omics comparisons between tox-positive and tox-negative donors. And finally, this integrated information can be translated into actionable biomarkers to support stratification, monitoring, and clinical translation and safety finding. So together, these capabilities form a complete framework for DILI decision-making. They allow early detection of toxicity risk, mechanistic understanding of liver injury, and identification of susceptible patient populations. By benchmarking compounds against reference DILI drugs, results are placed in a clinically meaningful context that supports confident go, no-go decisions. And ultimately, this improves predictivity and helps reduce late-stage attrition in drug development. So thank you very much for your attention. You can follow us on LinkedIn, and if you have any further question, you can also send us your question in your emails. And we are open also for the question during this webinar.
Thanks so much, Sarah.
All right, great. So hi, I'm Vignesh, and primarily I think TissUse is known for multi-organ chip. So today I would speak about a human-make liver plus X chip platform. So what are the combinations possible? And briefly touch about the liver ring trial and key findings from there. And so just to give you a bit of an introduction about TissUse, so we were spun off from the Technical University in Berlin. And from the very early on, we were focusing very closely to work with our partners in industry and academia after a couple of years to work for fit-for-purpose assays and developing different assays that will be relevant for them. And then later on transferring this to these partners and then supporting them with providing chips and different equipments. And in general, if you look at how TissUse is working, so in general, from the very beginning, our focus has been on body on a chip, so going towards full body on a chip. But so far, I think most of our publications have focused on the multi-organ aspect. But it's also possible with TissUse platform to do single organ on a chip and build towards a multi-organ, for example, liver plus X, as I talked earlier. And there's a huge benefit for this like kind of workflow. Essentially, from the very beginning, if you look at it at TissUse, we are focused on different chips and devices and assays that we have developed for our clients and establish these different biological models based on different contexts of use. And then further on, then you can pair it with another organ. For example, as you see here with liver, kidney, or liver, lung. And then afterwards, these were transferred to our industrial partners. And then for these specific use cases, we also have about 17 different context of use assays, which are at a higher order, which means that not only were they working in our labs, but also working in our partner labs as well. And further on, some of these are now also included in different R&D submissions as part of different data packages. And also, we are also part of the earlier talked about I-stand validation pathway through CDER as well. And in general, if you look at it, I think early on, we also had a bit of talk about personalization of platform. This is also something we also do. Essentially, the most complex organ modeling that is possible at TissUse is a 4-organ chip. You can, from a single IPS line, differentiate intestinal model and also seed the individual channels with blood circuitry and also liver spheroids and kidney, and then add either brain, skin, or cardiac spheroids. And essentially why we do from the very beginning multi-organ is that we found that the organ-organ crosstalk is super important for disease modeling or in general as well. We showed, I think around in 2017, that when we did liver islet co-culture, that the liver is important for regulating the insulin and glucose levels. And also, we tried to maintain the physiological fluid tissue ratio as well in the chips as well. And there is also a lot of possibilities to also compare, because one of the key use cases for multi-organ is to replace animal models. So a lot of our initial clients were also asking us to compare between animals on chip and human on chip, and we showed it in a couple of examples there as well, comparing thyroid liver co-culture in human versus rat. And all of these were actually moving towards more predictive models and insights for our clients, and these also generate a lot of publications as well. And largely, some of them have also been adapted by major companies for advancing their preclinical research. I think today, the most important focus, I think we also heard about this a bit earlier, in general, for past 5 years, we have been focusing more on the regulatory side to see if we can go on qualifying some of these models that have been there. And I think one of the key indicators was this survey, which identified the most important problem was confidence in the data. And then we asked ourselves the question, how can we increase the confidence? And naturally, we came to a liver ring trial, and we wanted to do this cross-company pre-qualification of our liver model and increase confidence for regulatory submissions and tackle the most important aspect of reproducibility and robustness of the model. I think this was a very challenging project in general. It's still ongoing in some sense, and because of that, I can't share a lot of key data today, but I can give you some key insights, key learnings about how to do these kind of ring trials, essentially. Before we dive into that, I would like to briefly just describe our HumanMake Chip 2, which we use for this purpose. So it's a standard chip with PDMS based microfluidic membrane. At the bottom, you have a glass slide, so it's possible to image them. And it's a very dynamic system, which allows tissue-to-tissue communication. As you see here, the 2 separate culture compartments are connected. And on this, we have this on-chip micropump, which enables this pulsatile flow, which is super important for long-term cultivation of cells. And it's also possible in a single chip to have 2 independent circuits. So you can have 2 separate experiments in the same chip. And as you see here, it's possible to have that you have in one compartment organ A and connect it with another compartment organ B. And what this essentially means, and what we have shown in some of our publications, is that we are able to show different forms of liver spheroids that are possible. One is directly on the glass slide, or in these micro-engineered cavities or DINARE, where you can have individual spheroids there, or in hydrogel, like how you see here on the left, or combined together with different kinds of models, either skin model or another intestinal model as a barrier model example, or as a scaffold, as you see here for the bone marrow model in the center, or as an organoid model for kidney. And essentially, this really shows the usability to first develop like for some mature organ model like liver, and then further on qualify them for other co-culture combinations. Essentially, we also keyed in on this DINARE model, and the reason for that was that there is not much difference between stellate cell outgrowth between the on-glass slide model and inside the DINARE. So in the DINARE cavities, the difference between different culture compartments, what was seen in one paper, was actually much more stable. And that's why increasingly we went into this direction. And this is the one that we use for our liver ring trial as well. And going further into that, so the liver ring trial was just a single liver on a chip model. And these were done on these microvalve cavities. What you see here in the liver compartment, each cavity had individual spheroids, and they were also coated with ultra-low attachment coating. This is something that facilitates the cell growth there. And you have about 200 cavities there. So technically, you can have 200 spheroids, but essentially, we had between 50 to 100. And each of these aggregates about 500 cells. And mainly, I think the key question is like, what are different endpoints that are possible and how long can we do it? So the model so we divide usually into 3 phases. One is the model preparation phase, which can be between 1 to 4 weeks. In this case, it's seeding the cells directly in these cavities and the spheroid formation. And after that, once they are at the right stage, then you can do your studies. And essentially, the key endpoints during this was metabolic analysis through LC-MS proteomics. And we also look at glucose and lactate, and also viability through the samples that are collected during medium exchanges. And in the very end, it's also possible to do different histology as well. So there are already some publications from us on liver plus like another organ model in co-culture on our chips. It's also possible to do 4-organ as well. As I described earlier, everything from the same iPSC background as well, and also different cell sources as well. But today, the focus is on primary hepatocytes, which was done in one key paper. These are different liver models that are established already at TissUse, and different functional readouts that have already been done. And I think the most important aspect is that we are not limited with our own in-house models, but it's also possible to work with commercial liver models as well in our system. I think the key difference is that we have to again validate them for the same different readouts as we have done here, different commercially available human liver models directly adapted into the chip, and then the same readouts were also looked at. And as they improve, it's also possible to integrate them in our system, not just work with our in-house model as well. Going into the ring trial, I think it was a very large consortium and a multi-year project. So we were, of course, the key tech provider. Apart from that, we also had excellent modeling team from ESQ Lab and MPS Labs, who were part of the digital twin liver MPS of the data that was generated with the ring trial on these chips. And we had 6 amazing industrial partners, and also Texas A&M University, who are part of it as the core cohort. And bulk of the work was actually based off a paper from USB Pharma, so that's why they were also the leading company here. We also got guidance from different regulatory bodies here in Europe who are guiding us about setting up the whole project as well. I earlier alluded to the paper. So this paper was performed from Benoit Cox in 2022, and it was looking at different liver on a chip platforms and comparing it between plate-based and MPS platform and individual liver spheroids. And in this paper, already they looked at the viability, LDH for viability, and they also looked at individual markers like albumin and urea production. It also already looked at also a single diclofenac metabolism as well, comparing between liver chip and spheroid culture and also control. So already a lot of work was established for us from this paper and this model that was done at UCB Pharma. And they also looked at also encapone and tocapone DILI versus non-DILI compounds as well for safety assessment as well. And this is just the one example that I can show, which is blinded about DILI and non-DILI classification was done for the liver ring trial. Unfortunately, I can't share the whole unblinded list right now because it's going to get published soon. And until then, this is just a good indicator about how things were done. And just to give you an idea about the different arms of the liver ring trial, so we had essentially two arms. One was the DMPK arm and one was a tox arm. On the DMPK arm, we had a single study where we tested these 4 compounds that you see here on the right. And for the tox arm, we had 2 separate arms with 20 Cmax and 5 Cmax about these compounds. And I think the current trend in the field is to have a clear context of use for a specific assay. But for the ring trial, when it was set up, there was no clearly defined context of use. The primary objective was to essentially develop and qualify the standardized liver model that was established already before and show that it's really reliable and reproducible across different laboratories. And I think another key learning was the selection criteria for the compounds. And I think here we looked very closely at the compounds that were binding to the PDMS membrane in the human CHIP2. If there was a lot of binding, these compounds were not selected. If there wasn't much binding, then they were selected. So non-specific binding test was very critical about selection of these compounds. A lot of them were selected from the IQ MPS list of reference compounds for qualification of liver MPS. And selection was based on the PD profile after LCMS measurement. And here we also see 2 compounds alone, which was rejected, and other which was actually accepted into this. So if it had a high log P value and it's binding to the PDMS, it was then not selected because it's very hard to then replace it. Essentially, if you look at the experimental view, each of the companies who are part of it and also the other testers, they were provided with 2 human starters. So essentially, so you had about 16 circuits per experiment, and the test items were assigned using a randomized block design. And the design principles of this was earlier described in a Cranes paper from 2023, and that was the basis for this. And then the control units were distributed across different starters to address control unit variability and across chips to account for chip-by-chip variability. I think the most important learning that we had was on the donor sourcing, and this was I think the most critical thing for anybody else who would like to do similar liver ring trial, that the potential variability has a great impact on the data that comes out of the liver trial. And TissUse and UCB looked at multiple hepatocyte donors and evaluated for suitability and best performing donor. And only after that, we selected them and sourced to all the different partners in the study. I think the key things that we learned was that the donor source has to be from the same vendor, and the media and FBS lots, everything was sourced from the same vendor. And this was very important for the performance. Everything was ordered centrally by TissUse and allocated and distributed to all the partners. And the partners then performed the study, and then afterwards the compounds were sent again to TissUse, and then it was then further shipped to different other partners for carrying out the analysis, and finally also the data analysis as well. Another key thing was training, because a lot of...
Sorry, just one more minute.
Okay. Another key thing was training. We realized that the training was super important. If some participant later on was not able to take part in the training or didn't have the training, we saw that that particular cohort had a variability in the study as well. And currently, this is the status of the study so far. And so you can expect a manuscript from the consortium sometime in Q3 in 2026. And yeah, I look forward to further questions from you later on. And thank you for the other participants as well.
Thanks, Vignesh.
Thank you, everyone, and hi. My name is Riya Sharma, and I'm a senior scientist in the Research and Development Department here at STEMCELL Technologies. And today I'm going to talk to you about hepatic organoid systems and how you can use these versatile, complex in vitro models to gain tissue-specific toxicity insights in the effort to advance drug discovery and basically use them as a new application methodology. So I'm sure most people are already familiar with what organoids are, so I'm not going to go too in-depth into an introduction, but basically over the past decade and a half, organoids have emerged and continue to evolve as a revolutionary new tool for in vitro research. And these 3D in vitro cell culture models are essentially made up of multiple organ-specific cell types that are spatially self-organizing through cell-cell or cell matrix interactions to resemble the key phenotypic and functional features of the tissue of origin. And since the reemergence of this technology in around 2009, with the study by Sato et al. Describing a 3D expandable organoid model of the mouse small intestine, several complex organoid models to represent basically almost all the tissue types within the body have been developed. And so organoid models present an incredible opportunity to advance both basic and translational in vitro research, where currently used systems may present certain limitations. And these patient representative organoids present specific advantages because they can be expanded, banked, and used to recapitulate a diversity of donor phenotypes, genotypes, and backgrounds. And this is overall allowing all of us to leverage the biological complexity of these models for mechanistic disease and toxicity investigations, to study the compound efficacy and tissue-specific toxicity evaluations, as well as apply them for novel target discovery. And all of this collectively is contributing to an overall enhancement of experimental predictivity in a wide range of applications across the drug discovery process. So before moving on to some specific application examples that I want to show you today using hepatic organoids, I want to provide this very high-level overview of workflows that have been developed over the years to generate, maintain, and use organoids for various applications. So as I mentioned previously, the starting material for these cultures can be quite varied. So you can start with either fresh patient samples, you can work with pre-established organoid lines, or you can use pluripotent stem cells. For tissue-derived organoids, you're first preparing samples by isolating the appropriate cell type of interest from the tissue, versus for pluripotent stem cell-derived organoids, you're first differentiating those PSCs into the appropriate lineage using specific protocols that are optimized to the tissue you're working with. Once your starting material is processed and acquired, you're then culturing this in an appropriate cell culture medium that is supporting the expansion and in some cases differentiation of the stem or progenitor cell populations to form organoids. And this can be done either in a matrix embedded format, so using extracellular matrices like Matrigel, for example, in dilute matrix containing suspension. Once your organoid cultures are established, in many cases, these are then also compatible for long-term regular passaging and scale-up and cryopreservation, so allowing you to biobank your donor samples. For some tissues, including the liver, organoids can be differentiated further to then generate a more representative cell population and function, and to transition them to particular culture formats that might be more conducive to the desired assay. So in identifying tissue-specific toxicities, different assessments will require different model characteristics, both biological and practical. And so leveraging these tissue-specific differentiation potentials of cells grown as organoids allows you now to flexibly maximize the biological relevance of a wide range of experimental setups. And some of the differentiated culture setups are shown here, including just maintaining them embedded in matrix domes, or opening the organoids up to access both the apical and basolateral surfaces using a monolayer culture, constructing more tissue representative co-cultures with more complexity, as well as applying the organoids as a proliferative epithelial cell type for use on organ-on-chip or MPS applications. So this audience is obviously very familiar with the importance and value of liver models that are accurately recapitulating the maturity and function of the human liver. And traditionally used liver models, including primary human hepatocytes, immortalized cell lines, and non-clinical animal models, have been successfully used to help us understand the liver and its function for many years, but are also presenting certain challenges, including rapid dedifferentiation, as was highlighted earlier today, as well as loss of viability, or an incomplete representation of the metabolic maturity of the liver itself. So because of all of this, there's still a need for something that is scalable, remaining functionally relevant, patient representative, that is also compatible with longer term studies. And this is the need we're hoping to address with the human hepatic workflows that I'm going to talk about today. So the first of these is shown here. This is the Hepatocult Organoid Kit Human, which is designed for supporting the establishment, expansion, and differentiation of hepatic organoids that are derived from patient liver tissue samples. And these proliferative intrahepatic cholangiocyte organoids can be consistently established across patient samples. So we've had 100% success rate with every tissue sample we've worked with. They can be expanded long-term, as shown in the graph on the top, with consistent exponential growth. They can be cryopreserved for future experimentation and biobanking. And they can also be differentiated to generate hepatobiliary organoids that are exhibiting key characteristics of mature adult hepatocytes. And shown here is some representative CYP3A4 activity data in response to modulators like calcitriol and ketoconazole, which induce and inhibit CYP3A4 activity, respectively. The second workflow is the stem-diff hepatic organoid media, which includes a growth medium and a differentiation medium to support the generation of hepatic organoids from human pluripotent stem cell-derived hepatocyte-like cells. And I'll go through the workflow in the subsequent slide, but essentially these organoids are also expandable and compatible with long-term expansion, and then can be cryopreserved for biobanking. They can also be further differentiated to generate a hepatobiliary organoid culture that is exhibiting key features of mature hepatocytes. So taking a minute to review the workflow for this PSC-derived hepatic organoid culture system, which is going to be the focus of the remainder of these slides, we're essentially starting off with, excuse me, pluripotent stem cells that are first differentiated in 2D over 21 days to generate hepatocyte-like cells that are still retaining some fetal characteristics. However, if we take these hepatocyte-like cells, mechanically dissociate them, and embed them into extracellular matrix, we can generate these proliferating organoids that can be expanded on a weekly basis for several passages, as shown in the growth curve on this slide, and potentially also cryopreserved if you want to generate a bank of multiple donor samples. In this proliferative state, you're essentially able to scale and build your donor bank profile. But if you don't want to be generating these cells from scratch from pluripotent stem cells every time, we've also recently launched cryopreserved organoids for sale. So these are human iPSC-derived hepatic organoids, and they're essentially cryopreserved fragments from pre-established and qualified HLC-derived proliferating hepatic organoids. And on your end, they're ready for immediate expansion and scale up post-thaw, and then they can be downstream cultured and differentiated for the use in assays. And this differentiation protocol looks like this. So essentially you start off as if you're seeding a regular expansion, but then over the course of the next 17 days, through a series of media changes, you can differentiate these cultures to yield differentiated organoids that look something like what's shown on this slide. And now these differentiated organoids are ready for use in various applications. So I'm going to go through some characterization data to speak to what the phenotype and genotype of these differentiated organoids is in subsequent slides now. So if we characterize these human pluripotent stem cell-derived hepatic organoids, we see, once differentiated using the hepatic organoid differentiation medium, they're now expressing several key mature hepatocyte proteins, including albumin, alpha-1 antitrypsin, as well as BSEP. And you can also see expression of the hepatic transcription factor HNF4 alpha pretty uniformly across most cells. Some organoid regions also retain expression of hepatic progenitor or ductal markers, such as alpha-fetoprotein or started 19, suggesting a bit more of a hepatobiliary phenotype. These differentiated PSC-derived hepatic organoids are also secreting increasing levels of serum protein albumin over time into the spent medium, and they're exhibiting measurable CYP3A4 activity, which can be reproducibly modulated in a dose-dependent manner across donors using compounds like ketoconazole, which is an antifungal known to inhibit CYP3A4 activity, and canceltriol, which is the active form of vitamin D and known to induce CYP3A4 expression, leading to an induction of activity. So these three data points are indicating that the differentiated PSC-derived hepatic organoids are a good model of key hepatic functions, providing a basis for a non-cytotoxic assay for hepatocyte functionality. And I'm now going to go through some examples of this application. Starting with the use of these PSC-derived liver organoids for the detection of specific compound toxicity in a short-term drug exposure assay. So this is over a period of 72 hours in the presence of compound. So here we treated the organoids with triglyceridone and nifazodone, both of which are compounds with reported high DILI severity class, and they're shown in the darker orange and the darker gray lines here, or also treated with pioglitazone or buspirone, which are related compounds, or in the case of pioglitazone, a structural analog of triglyceridone. And these are known to be less toxic with a lower DILI severity class. And these are shown in the gray and lighter orange lines. So what you can see is that the hepatic organoids are showing a dose-dependent viability response and are exhibiting differential sensitivity, allowing us to distinguish between compounds with different DILI severities, indicating that these models detect specific toxicities. Similarly, these PSC-derived liver organoids are also compatible with long-term differentiation and culture, thereby enabling their use in long-term compound exposure assays. And this is useful for the assessment of chronic or delayed toxicity. So in this particular experiment, the organoids were differentiated for over a month and treated with related compounds phenyluridine, which was found to have delayed onset hepatotoxicity in patients, and its non-toxic analog 2-deoxy-2-fluoro-5-iodouridine, also known as InSphero, and this treatment was done over a period of 4 weeks. An assessment of viability over this period of time then demonstrated to us that these hepatic organoids were in fact able to model the progressive and time-dependent toxicity of phenyluridine and actually capture the difference in toxicity between the two analogs. We're also continuing to expand on these tox applications further, and one such application is the modeling of compound-induced cholestasis, which is the last application I want to talk about today. So cholestasis refers to impaired bile flow and can be a contributor to drug-induced liver injury. And to run these assays, we use the molecule CMFDA, which is 5-chloromethylfluorescein diacetate, and this is cell-permeant and non-fluorescent, entering the cells passively. Once inside the cells, it's cleaved by cytosolic esterases to produce membrane impermeable and now fluorescent 5-CMF or 5-chloromethylfluorescein. And this 5-CMF now has to be actively effluxed by transporters BSEP and MRP2 out of the cells. And this whole mechanism is allowing us to visualize bile transport and potentially bile canalicular structures. In these particular organoids, we expect the CMFDA to enter the cells, forming the organoid, and then to be effluxed either into the organoid lumen or back into the culture medium. And if we treat these organoids with bile efflux inhibitors or cholestatic compounds, we'd expect the 5-CMF signals to remain localized to the intracellular compartment rather than be effluxed into that lumen or spent medium. Since this assay relies on imaging-based readouts, I also want to highlight another tool here, which is the 96-well plate organoid culture plates. So these are a newer culture plate format that are replicating the typical approach to organoid culture, but instead of a curved dome, we're culturing them in a flattened plug with a defined lower thickness than a typical dome, and including a step feature to eliminate the meniscus. And this overall is allowing us to significantly improve and simplify imaging, culture uniformity, and handling in both manual and automated workflows. So getting into the results for these cholestasis experiments, when we treated cultures with triglyceride and cyclosporin, which are both known to inhibit bile efflux, we're observing higher green intensity within the organoid cells, and this is corresponding to the inhibition of the efflux of the fluorescent 5-CMF. And this increase in 5-CMF-related intensity is dose-dependent, as seen in both the fluorescent images and the quantified fluorescent intensity data right underneath, which is normalized to % confidence of organoids. And the green intensity in both the triglyceride and cyclosporin A conditions is in fact significantly higher as compared to the matched vehicle control or the untreated controls, while pioglitazone, which was the structural analog of triglyceride, which is not reported to be cholestatic, is not inhibiting 5-CMF efflux as evidently, and organoid fluorescence intensities in these cultures are more closely resembling the vehicle control in both the images and the quantification. So the organoids are responding to bile efflux inhibitors and are able to distinguish between cholestatic and non-cholestatic drugs. And in vitro models like these organoids can be used not only to detect sensitivity to compound-induced DILI, but also to start to explore the mechanism that might be contributing to that detected toxicity. So that concludes the data I'm showing today, but we have several other pieces of application examples that I'm happy to review with you another time. But overall, I wanted to highlight the application of these hepatic organoids as complex in vitro models of the liver, and just wrap up by summarizing some of the benefits of stem cell-derived models, such as organoids, as high-fidelity predictive tools for liver research. So these organoid models are allowing you to reduce inaccuracies due to interspecies differences while increasing translatability of in vitro results to human clinical outcomes. Organoids are derived from cells that are better reflecting the biological phenotype, healthy or diseased, and the cellular composition of the tissue so the liver. They're expandable and bankable, allowing you to model patient and population-specific responses, but also allowing you to proactively manage donor-to-donor variability through the establishment of standard lines and panels that you can repeatedly use. And lastly, organoid-derived cells can be assayed in a wide variety of formats, including monolayers and also organ-on-chip or other microphysiological systems. If we have questions, time for questions now, I'm happy to take them, but otherwise, thank you so much for your attention, and please feel free to reach out with any questions that you might have after the webinar. Thank you.
Thank you, Ria.
Okay. Good afternoon, everybody, and thank you for the opportunity to speak today. My name is Muya Lee. I'm the founder and CEO of Bioprinting Labs. Today, I'd like to introduce our scalable pillar precision plate platforms for liver organoid culture and discuss how it can support high throughput compound screening, disease modeling, and non-animal testing applications. So as you know, traditional 2D cell culture and many static organoid culture lacks physiological relevance and also scalability for predictive drug screening. Static culture often suffer from poor nutrient transport, necrotic core formations, and limited reproducibility. To address these limitations, we developed pillar precision plate platforms, which integrate 3D bioprinting, dynamic precision culture, on-plate cryopreservations, and high-throughput screening into a single workflow. Our goal is to provide a scalable and assay-ready organoid platforms compatible with existing pharma screening infrastructures. Traditionally, organoid has been cultured using matrix domes, ultra-low attachment well plate, spinner flasks, and the microfluidic systems. However, these platforms often face important limitations, including low throughput, poor reproducibility, necrotic core formations, difficult handling, and limited compatibility with the automated screening workflows. Our pillar precision plate address these challenges by enabling scalable, dynamic, and assay-ready organoid culture in a standard created for footprint. These are our core products. We currently provide injection molded pillar and perfusion plate in 36, 144, and 384 pillar configurations. Importantly, these products are fully compatible with the standard lab equipment and screening system already used in the pharma and biotech laboratories. No dedicated pumps or specialized instrumentations are required. This slide illustrates our 144 pillar plate and the 144 perfusion plate systems. Cells and organoids in hydrogels are loaded onto the pillar, while the perfusion plate contains wells and reservoir connected through microchannels. When the two plates are sandwiched together, the system supports dynamic bidirectional perfusion cultures. This design enables continuous nutrient exchange and improved long-term organoid maintenance. When the pillar plate are placed on the digital lockers, periodic tilting generates forward and backward flow. The resulting flow rate are approximately 5 to 20 microliter per second, which are similar to microcapillary flow conditions. Importantly, these pump-free perfusions improve nutrient transport and oxygen delivery while maintaining a simple and scalable workflow. Cells and spheroids encapsulated in hydrogels can be loaded onto the pillar plate using multiple different ways. These include manual cell loading and cell dispensing, pillar plate stamping, spheroid transfer from ultra-low attachment well plate, and the microarray 3D bioprinting. This flexibility enables rapid and reproducible organoid manufacturing for screening applications. After cell loading, the pillar plate can be integrated with the Petri dish, deep well plate, or perfusion plate. This allows users to perform both static and dynamic organoid culture workflow using the same platforms. This workflow is simple, modular, and compatible with existing lab practices. One important advantage of our platform is that organoid can be stained, imaged, and analyzed directly on the pillar plate without harvesting. This significantly simplifies high-throughput imaging workflows. Whole mount immunofluorescence staining and imaging can be performed by attaching a thin glass slide beneath the pillar plate. And the pillar perfusion plate is fully compatible with common lab equipment, including fluorescent microscope, plate readers, and automated imaging systems. This eliminates the need for additional capital equipment investment and facilitate immediate integration into existing workflow. Uniformity of cells is critically important for reliable compound screening. Here we demonstrate highly uniform spheroid and organoid formations on the pillar plate across multiple tissue types, including liver organoid and tumor spheroid. This uniformity improved assay reproducibility and the data reliability. We have applied the pillar perfusion plate platforms to generate a variety of different organoid models, including liver, brain, intestines, cardiac, and tumor organoid. These applications has been validated through multiple peer-reviewed publications over the past few years. If you are interested in the paper published, please have a look at this company website. Our platform provides several key advantages. First, scalable pillar plate stamping and the microarray 3D bioprinting enable rapid and reproducible organoid generations. Second, pump-free dynamic perfusion improve the nutrient transport and organoid maturation. Third, the standard 384 well format support high throughput screening compatibilities force assay miniaturizations significantly reduce reagent and cell consumptions. And finally, on-plate cryopreservation support standardized organoid manufacturing. Now I'd like to focus specifically on liver organoid applications using our pillar perfusion plate platforms. I will highlight scalable liver organoid generations, dynamic hepatic maturations, hepatotoxicity testing, liver regenerations, steatosis modeling, and cryopreservations of organoid. Here we demonstrate the generations of expandable liver organoid derived from iPSCs. These expandable liver organoid can be passaged multiple times and provide scalable source of organoid for drug screening applications. This slide demonstrates microarray 3D bioprintings of cells and spheroid suspended in hydrogels onto the pillar plate. These processes enable scalable and high throughput, highly reproducible organoid manufacturing compatible with the high throughput workflow. Here we demonstrate 3D bioprintings of cells and spheroid in hydrogels onto our pillar plate. Organoids suspended in the hydrogels can be loaded in the disposable tips, and the machine can pick up these samples and then directly print it on the pillar plate. These processes ensure uniform loading with the coefficient of variations of 10 to 20%, enabling highly reproducible organoid productions at a scale for drug screening. And after screening, imaging can be also done in a high throughput manner. As you can see, we can utilize this automated fluorescent microscope to get the images. And after imaging, the cells can be cultured in the dynamic conditions in a CO2 incubator. Here we evaluated the reproducibility of 3D bioprinting on the pillar plate. Cell viability after printing remained very high, close to 100%. In addition, organoid loading showed low pillar-to-pillar variability with the coefficient of variations approximately between 10 to 18%. These results demonstrate robust and scalable organoid manufacturing. Using expandable liver organoid printed on the pillar plate, we successfully cultured liver organoid under both static and dynamic conditions. Dynamic perfusion culture supported improved nutrient transport and long-term maintenance of liver organoid. Here we compare the hepatic maturations and functions between static and dynamic culture conditions. One represent static organoid culture, and the two represent dynamic liver organoid culture. Dynamic perfusion significantly enhanced the hepatic gene expressions, including markers such as albumin, CYP3A4, UGT1A1, SULT2A1. We also observed increased albumin secretions under dynamic conditions. These findings suggest that perfusion improved hepatic maturations and functionality. Immunofluorescence staining confirmed expressions of key hepatic epithelial and hepatocyte markers, including CK18, albumin, and E-cadherin. These data further support hepatic differentiations and maintenance of epithelial structures within the liver organ. We next evaluated acetaminophen-induced hepatotoxicities using human liver organoid cultured on the pillar plate. Following treatment, we generated quantitative dose response curves and determined IC50 values using standard ATP-based viability assays. These results demonstrate that the platform's utility for predictive toxicology screening. In addition to toxicity testing, we also evaluate regenerative recovery following acetaminophen-induced injury. Inflammatory gene expression increased after injury and subsequently decreases during organoid recovery and expansions. These findings demonstrate the potentials of liver organoid for studying liver injury and regenerations. Here we induced the steatosis in the liver organoid using oleic acid treatment. Lipid accumulations was visualized using body P staining, demonstrating the platform's capability for fatty acid disease and NASH modeling. Confocal imaging and quantitative analysis confirmed significant lipid accumulations following oleic acid treatment. These results demonstrate that platforms support both imaging-based and the quantitative analysis workflows for liver disease modeling. Finally, we evaluate on-plate cryopreservations using dynamic perfusions of cryoprotective agents. Cryoprotective agents can be flowed within the perfusion plate, while the spheroid and organoid can be encapsulated on the pillar. So dynamic CPA perfusions improve post-thaw viability of spheroid and organoid following ultravitrifications in the liquid nitrogen vapors. This capability may support standardized organoid manufacturing, storage, and the distributions. To summarize, our platform provides 4 major advantages. First, precision and reproducible organoid manufacturing. Second, physiologically relevant dynamic perfusion culture. Third, on-plate cryopreservations. And lastly, integrations with the existing pharma screening infrastructures. Importantly, these platforms require no specialized instrumentations and can be rapidly adapted within the existing workflow. And thank you for your attention. If you are interested in collaboration, beta testings, or additional information, please feel free to contact us or visit our website at 3dbpl.com. I'd like to answer any questions you may have. Thank you.
Thanks, Muyil.
This is Alex Salmonen. Awesome. I manage the in vitro assay services arm of LifeNet Health, but today I'm going to be speaking on behalf of LifeNet Health Life Sciences to showcase some of our recent work in the liver MPS space. So thank you for having me. So specifically, I will be talking about the LifeNet Health MPS and how we are utilizing an integrated multi-organ system approach, so maybe a little bit different from some of the talks we heard today, to study drug-induced liver injury and ADME. The LifeNet Health MPS platform features a standard 6-well plate format, as you can see here, and this is with individualized wells interconnected via what we call a simulated vascular system. So this vascular system is comprised of a series of semi-permeable membranes that navigate through the culture wells to allow for exchange of small molecule drugs, in many of our cases, nutrients and signaling molecules like cytokines across organ models. This allows us within the single test system to capture whole body ADME, to study secondary and downstream toxicities, including hepatotoxicity, of course, it's the scope of my talk today, and ultimately improve prediction of human relevant exposures. So what makes this platform stand out and sort of facilitate these complex in vitro kinetic studies is, to me at least, its simplicity. So shown here is a 3-organ configuration of the MPS comprising an intestine, liver, and kidney culture systems. The simulated vascular approach, again, allows for physiological exposure without compromising culture conditions. So these cultures are all maintained in their own medium, and minimal to no fluid exchange means that we can ensure that the cultures are healthy and compatible with your standard clinical toxicology endpoints. It is also worth noting here that we are working with a blood velocity that is comparable to the velocity in the capillary beds in vivo in humans. So critical fluid dynamic parameters such as the competition between infection and diffusion are conserved. And this is a very important point to make when translating these data to an in vivo PK prediction specifically. A typical output from a study utilizing the LifeNet Health MPS platform includes pharmacokinetic and toxicokinetic data. Shown here is just an example of some of the work we performed with the US FDA's Human Foods Program, and which you can see is dose-dependent kinetics of acrylamide, which, for those unfamiliar, was a compound of a rich history with the Human Foods Program and particular interest to them. We additionally observed toxicity endpoints comparable to those seen in the rodent studies as a consequence of these acrylamide kinetics. So you can see here, and this includes within the liver. And while acrylamide itself is a pretty well studied compound at this point, this multi-organ approach to evaluate DILI and of course, ADME is critical for compounds that have unknown first-pass metabolism or intestinal effects that may result in downstream hepatotoxicity. And I just want to highlight again that these endpoints, LDH and MTT for the intestine, LDH, ATP, and glutathione for the liver, and then beta-NAG and Chem1 for the kidney are all clinically relevant endpoints, which are observable in this platform for each tissue due to that individualized simulated vascular integration approach. And importantly, we're not limited to these endpoints seen here, as we routinely evaluate additional biomarkers like cytokine production, urea and albumin, and even gene modulation through RNA isolation and qPCR. So as we continue to expand on the use cases for this platform, the individualized culture well format has shown to be of particular benefit as it permits wide adaptability to a variety of established, in some cases, such as skin and ocular OECD compliant culture models. So we can model, say, inhalation exposure with epi-airway or epi-alveolar models from Maltech. We can model oral exposure with small intestinal models like epi-intestinal, of course, KICO. Two cultures, skin or dermal exposure with models like melanoderm and epiderm from Maltech. And then really what I want to focus a little bit more on today is the application of more advanced hepatic models such as TruVivo. So TruVivo itself is a all-human 2D hepatic culture system. So it's comprised of primary human hepatocytes, stromal cells, and endothelial cells. In this platform, they're co-cultured and they self-assemble into this tissue-like organization. Native cell-cell interactions and architecture are produced, and we have stable morphology and hepatic function as well. And finally, you can actually see stable biocanalicular network formation, which can be very important for applications like cholestasis, as you can see here. LifeNet Health has a unique position in this space as we actually control every step of the model production process for TruVivo. As a federally designated organ procurement organization, we facilitate organ and tissue procurement, which are then transferred to our experienced cell isolation team. Following isolation of these key cells like hepatocytes, stromal cells, and endothelial cells, we extensively characterize the hepatocyte specifically for compatibility with the TruVivo culture system, as well as compatibility of TruVivo with the MPS, catering to, of course, the context of use. It's also worth noting that as the tissue source ourselves, LifeNet Health, we have access to extensive donor medical history for these hepatocytes, which can provide insight into sort of disease morphologies. And all hepatocytes undergo additional characterization for genotyping, so 112 SNPs there, as well as phase 1 and 2 enzyme activity and CYP enzyme induction, as we've seen quite a few times today already. All of this translates to sustained hepatic enzyme activity over 4+ weeks in the TruVivo culture system. And tying this back to the scope of this talk, this gives us increased confidence in the relevance of clearance predictions, both in TruVivo alone, as well as within the MPS platform. So you can see here, just some select CYPs, 2D6, 384, and then UGT activity. We're seeing sustained activity over 31 days in the TruVivo culture system, which is a substantial improvement over maybe more classic ADME models like suspension hepatocytes. And so just to sort of end the talk here, I'm going to focus on a couple of case studies utilizing the MPS platform as well as TruVivo. The first one is an APAP case study we performed. In this case, we did a 20 millimolar APAP dose to the apical intestinal surface. So this is obviously modeling oral exposure and a 72-hour study. What you can see here is following the dose on the apical intestinal surface, you can start picking up acetaminophen, so the parent, as well as two of the metabolites that we focused in on here, the acetaminophen sulfonated version, as well as the glucuronidated version. In the basolateral intestine circuit, we can see it peaks, we get the Cmax, and then distribution into the liver, as well as the simulated vasculature or systemic or perfusate. Circling in a little bit more, you can see how we are actually generating first-pass metabolites in the intestine. Again, this is a very well-established epi-intestinal culture model that's just being interconnected in our culture system. We see further metabolite formation in the liver, and this generates this sort of metabolite profile that we would expect to see in vivo in serum. All of this translates to, of course, some sort of toxicokinetic response. Here we're seeing that compared to vehicle, this high dose of acetaminophen is slightly, but not substantially cytotoxic to the intestinal model, but we see significant cytotoxicity in the liver compartment, so in true vivo. So we're able to track both the kinetics, the metabolites, as well as the toxicity downstream of the intestine in this system. The second case study I want to talk about is just integrating true vivo and the LifeNet Health MPS for formulation development. In this case, we worked with a partnership at Bayer Healthcare to model essentially delivery of an API within what's called a self-nano-emulsifying drug delivery system, or SNEDs. So they wanted to evaluate a variety of SNEDs for application to protect their API. Again, we're using, in this case, the 3-organ MPS system with epi-intestinal, true vivo as our liver model, and then HK2 kidney cells. We're adding the API loaded in the SNEDs at various concentrations on the apical intestinal surface, and then monitoring via LCMS the API in the different compartments over time, as well as toxicity. And what you can see here is we're able to observe that the SNEDs loaded with various concentrations of API, so these are all the same concentrations of SNEDs, just different candidates, is pretty well tolerated. So we're seeing within our monocultures very, very promising results in terms of the safety profile of these SNEDs. And then when we move forward with a candidate SNED into the MPS platform, we're seeing this nice absorption and distribution of the API throughout the culture system, which is a promising result and supports favorable kinetics of the API when labeled in this, loaded rather, in this novel formulation. So these are the sort of outputs that we're kind of gathering for our clients here. And so pretty quick talk here. Maybe I'll have some time for questions, or at least we can answer some more later on. But I just want to summarize the LifeNet Health MPS and the True Vivo sort of collaboration we have right now. So the MPS itself is a standardized, again, well plate format that supports adaptable, validated tissue models. It's organ-specific media, enables relevant toxicity evaluations without the need for custom media development. And then the simulated vascular approach enables tissue-to-tissue communication, really while preserving culture conditions. And then, of course, being this liver focus here, the True Vivo integration into this MPS platform really extends the platform's use towards more physiological relevant multi-tissue insights and, of course, translational potential. So I hope these points came across well. And so with that, I'm just going to leave this up for a second. I'm happy to take questions if there's time. If not, we can save them for the Q&A. I also have a QR code there that links to some of our content libraries, which will give you some more explanations of our offerings there.
Thanks, Alec.
All right, great. Good afternoon, everyone. Thank you once again, Elisa and 3R team for inviting us. So here we introduce our work on iPS-derived liver organoid model utilizing VitroGel platform. So for the next couple of minutes, we'll explore how this fully defined xeno-free hydrogel platform serves a powerful foundation for advanced liver microphysiological systems. All right, so here at TheWell Biosciences, we provide a complete workflow for organoid culture, providing a fully integrated solution for 3D model establishment through downstream analysis. We begin with our VitroGel organoid, which is a xeno-free, 100% synthetic hydrogel system which support organoid formation and long-term culture. And then we have our xeno-free rocket cell culture media, particularly rocket cell organoid xeno-free essential core medium. This can consider as a universal supplement system to replace N2B27 and other small molecules. You just have to add growth factors to this medium, and you will have the complete organoid culture medium. And we also have premium growth factors. Together, we've come up with some liver media kits, which I'm planning to discuss a little bit more in upcoming slides. And then we also have our culture vessels, whether you are doing standard cultures or high throughput automation related work, our VitroPrime plates can be used. Then for endpoint analysis, organoid can be efficiently recovered using our VitroGel organoid recovery solution, which is an enzyme-free version, can be used for both synthetic gels like VitroGel or animal-based gels like Matrigel. And then we have downstream analysis kits such as Cyto3D Live Dead Assay Kit. Now, together, this workflow provides a standardized, CNO-free platform for developing robust liver organoid model for MPS, disease modeling, or drug testing related applications. So before discussing our liver organoid model, I think it is important to understand the unmet need in this field. So some I have listed here. So as we know, liver toxicity remains one of the leading causes of drug iteration, especially during development. And also traditional animal model often fail to accurately predict human-specific responses. While hepatic and 2D cultures are famous, sometimes we see this rapid loss of functions when you perform these 2D cultures, especially in vitro. Now, these all kind of could be a reason for inability to use them in long-term culture. So that is why it is important to have a better liver model. Right now, we have models, but there are several challenges we face with these liver organoid models. The one is batched variability, especially when you are using animal-based hydrogels, and also limited tunability associated with ECM systems, reproducibility and scalable challenges. And also when you are using animal-based gels, sometimes there are unknown components which might have a direct or indirect impact on your final results. So together, these challenges highlight the need for a defined, standardized, and scalable culture platforms, which can support the development of more predictive and human-relevant liver MPS models. So this is where the TheWell Biosciences come. To address all these challenges, we developed a fully CNO-free platform for generating iPS-derived liver organoids. And this approach actually combines key components, 3 key components. First one is our CNO-free hydrogel VitroGel organoid. And then we have our rocket cell CNO-free culture media, included with organoid CNO-free essential core media, and then our premium growth factors. Together, these components create a standardized, and the key factor here is animal component-free culture system for generation of functional liver organoids suitable for MPS type of research. All right, so this slide shows or highlights the fully CNO-free organoid development workflow we have at TheWell Biosciences. Starting with iPS-derived iPSCs, these cells are guided through several stages of the development, starting from definitive endoderm to hepatic endoderm differentiation, and then the early hepatic maturation, then finally mature liver organoids. Now, the key advantage of this workflow is that every stage that we have here is supported by defined and animal-free reagents. Now, during early stages of iPS-derived liver organoid generation, we use VitroGel stem and our rocket cell media. And when they are ready for maturation, we can transfer these liver organoids to VitroGel organoid hydrogel, which will support the synthetic microenvironment, which is required for long-term growth and maturation. All right, so moving on to characterization, In this slide highlights the cellular morphology of our iPSC-derived liver organoids. As you can see here in H&E staining, the cells within these organoids closely replicate the distinct cuboidal morphology, which is a characteristic of native hepatic parenchymal or hepatocytes. And also in this bright field image, you can see that which confirms the robust organized structure of these living cultures. And we can clearly see that cuboidal cell morphology formed in these liver organoids which are entering differentiation stage. Now here I have a bit more results to show how these liver organoids are developed, starting from spheroids till we get the mature organoids. And we have another line, organoid line, which is RFB label, could be used for co-culture type of experiments. And these data further shows during maturation, how this cuboidal structure formation can be seen. All right, so next, we confirmed the functional and lineage-specific biomarker expression of iPS-derived liver organoids using immunofluorescence staining. The first panel here, which shows the nuclear localization of HNF4 alpha, which is a master transcriptional factor, essential for hepatic differentiation, development, and functions. Then we also look at PCK2, again, an essential enzyme for gluconeogenesis and expressed in hepatocytes. And we see clear expression of PCK2 in our mature liver organoids. Then we also looked at SOX9 positivity, which indicates the presence of hepatic progenitor cells. And also SOX9 is important for bile duct formation and hepatic regeneration. And in our iPS-derived liver organoid model that we developed using our xeno-free platform, the SOX expression was clearly observed. Then the fourth panel here shows the expression of albumin, the hallmark functional protein synthesized by mature hepatocytes, and which is colocalized with the phelloidine, an outline of like the actin structure of these iPS-derived liver organoids. Now, together, these are some of the results, the markers that we tested. So together, this comprehensive marker profile confirms that our xeno-free system successfully drives both structural maturation and phenotypic functionality, which is required for an accurate liver model. Next, we evaluated the health and viability of our liver organoids using a live dead assay. On the left here, you can see how straightforward the workflow with our Cyto3D live dead assay kit. You just have to mix the reagent in media and then incubate for 10 to 15 minutes, and it is ready for imaging. And people who are interested, the protocol, you can scan the QR code to get more information. Now, these liver organoids are somewhat early, young hepatic organoids, just entering mature stage and differentiation stage. And we stain with Cyto3D live dead assay. The green indicates here the live cells, whereas in red, which are the dead cells. So these results confirm cell survival and structural integrity of our liver organoid model using this during this hepatic maturation. Now we have done this assay at different stages to confirm throughout this differentiation process, these liver organoids have cells are viable. All right, so to support this workflow from start to finish, we offer a comprehensive ecosystem for iPS-derived liver organoids. So these are some of the items that we're launching. First, our maintenance and expansion kit, which allows robust proliferation and long-term banking of your cultures. Then we have iPSC liver organoid differentiation kit that will provide precise chemical cues required for transitioning those cells into mature and functional hepatic lineage. Both these kits are included with xeno-free hydrogel. And then finally, we have iPS-derived liver organoids and also ready-to-use screen plates. These are delivering pre-cultured. These plates, the organoids are cultured in, and you can actually directly, we can send these to your lab to minimize the preparation time as well as maximize the screening efficiency. So with that, I would like to quickly highlight some key features and benefits of this full xeno-free platform. As I mentioned, all-in-one complete system. So basically, this optimized workflow and the kit eliminates the complex formulation steps, also possible calculation errors that you may have. And also the system is 100% synthetic and ready to use optimized workflow. Finally, this whole platform is scalable and automation friendly, which will maximize your efficiency in these liver MPS cultures. All right, so to wrap up, I want to leave you with 3 key takeaways regarding our iPS-derived liver organoid system. First, this model provides highly physiologically relevant liver structures and closely mirror native cellular morphology, which was discussed some of the data during my slides. Second, the entire platform powered by Vitrogel and rocket cell media, those are 100% xeno-free, chemically defined. And then last, our xeno-free system, including these iPS-derived liver organoids, can be immediately used for preclinical safety testing, drug discovery, or disease modeling type of experiment. I think that brings me to the end of the presentation, and I want to thank you once again for your time and attention, and highly encourage you to scan the QR code. Then you can explore how our end-to-end platform can support your specific 3D culture, and also follow us in LinkedIn. And if you have any questions related to this iPS-derived organoids or the liver kits, please feel free to email us via this email address. And yeah, thank you once again.
Thanks so much, Kalhara.
So yeah, thank you very much for the opportunity to speak here. So my name is Volker Lauschke. I'm a professor in translational pharmacology at Karolinska Institute, and today I'm speaking on behalf of Shanghai HepaBiotech, which we started around 2.5 years ago. But the company is based on the platform technology that we've been developing over the last close to 15 years by now. So we are a CRO that is based in Shanghai, in China, and our ambition is to develop 3D microtissue platforms for preclinical safety, efficacy, and metabolic assessments. And we have already, although rather young, we have already quite an extensive track record of working with leading pharmaceutical companies, not only in China, but worldwide. So our philosophy is that we're using a variety of different tissue models, all derived directly from primary patient samples. So this is all mature cells. It's not in vitro differentiated, not iPS-based or adult stem cell-based, but all fully mature adult cells that we are obtaining from tissue resections. We have a network of partnering hospitals also there in Europe and also in China, and are using those cells then to reconstruct these 3D human tissue models in vitro. And each one of those models is very extensively characterized. So we put a lot of emphasis on this integrative omics analysis, which means that we are taking one sample at the time of isolation of the mature cells. And this is our baseline, and then we're comparing our 3D tissue models over time time so that the models are accurately resembling the molecular configuration of those cells. And starting from healthy tissues, we've also established various disease models, so where we can take directly material from patients. So we don't have to induce the disease in vitro, but we just have to carry it over into the in vitro setting. And we focus predominantly on metabolic diseases, but also inflammatory and infectious diseases. We also construct and fabricate our own microfluidic devices, and this will be the central theme of today's talk, to then study the interaction between those different 3D microtissues. And lastly, we work extensively also on precision drug development. Essentially, since each one of those tissue models is coming from an individual person, this allows us to very early on, in a scalable fashion, identify already companion diagnostics or biomarkers that could identify, for instance, responders and non-responders, or individuals most susceptible to idiosyncratic toxicity. So we've established those tissue models from a variety of different human organs. Liver is the one that we worked on most extensively, but we also have adipose tissue, skeletal muscle, pancreatic islets, intestinal cultures, various solid tumors. But of course, today the central theme will be focused on the liver of my talk. So there's a multitude of different applications that we have already established. I will not go into much detail there. Hepatotoxicity prediction is something that we've worked extensively on. Also benchmark the system in multi-center trial together with both pharma and academic partners. Pharmacokinetic analysis, also use that extensively for IVIVE and PBPK modeling. We can use it for studying drug-drug interactions, importantly, not only those by the classical nuclear hormone receptors such as PX, RCAR, and AHR, but also non-canonical and more unconventional mechanisms that are usually missed. Again, I will not speak much about this in my talk today, and rather focus on the modeling of liver disease, and then also how dynamic interactions in a microphysiological system can aid these assessments. So very briefly, from the liver disease models, we have done a lot of work on fatty liver disease models, or metabolically associated steatotic liver disease, as it's nowadays called. Also a little bit on cholestasis and various viral hepatitis systems. And to jump into our mesh model, so fatty liver disease model, what we are doing here is we are taking cells either from control individuals or from patients with a histologically confirmed diagnosis of MASH, the more progressive inflammatory stage of the disease. We can then compare those. Already morphologically, those spheroids look clearly different. We can culture these for many weeks in culture, at least for 5. We can see that they are insulin resistant, the cells from those MASH patients, compared to the insulin sensitive cultures. And of course, now most importantly, we see that they are massively steatotic. So this is a confocal image staining for lipid droplets. Doesn't stop at the level of steatosis. We also see increased cytokine secretion. So this is true protein levels that are secreted by those spheroids. IL-6, IL-8, TNF-alpha, all highly significantly elevated. And we also see an increased deposition of collagen, which we can also detect in the supernatant using procollagen 1 secretion as a proxy. Importantly, this is another factor that sets our system apart from stem cell-derived models. And this is that we can use and co-culture our spheroids, which are not only consisting of hepatocytes, but also various non-parenchymal cells, and importantly, all the non-parenchymal cell species that are present in the liver at the time of isolation. So here I'm showing that exemplarily for the immune cells. Here we used a rather sophisticated FACS gating strategy that distinguishes the different immune cell culture model system, and now go a little bit into the MPS and the microfluidics. So in order to build our microfluidic devices, we've developed a new fabrication system called NanoRim, so nanoreaction injection molding. And this means that we are injecting a liquid monomer of stoichiometric tyrosine epoxy in our case into a pre-made mold structure, and then by purely shining on UV light, we can then cure these liquid monomers and produce in a very rapid and scalable fashion multi-scalar and multiplanar devices. So this means that we can, in a single production process, fabricate devices that are centimeter sized, but then also harbor millimeter-sized, micrometer-sized, and even nanometer-sized features, all in a single production process with very low clamping pressure and zero packing pressure, and importantly at room temperature. So this really massively scales up the possibility for parallelized fabrication. And so what is this material? So I briefly mentioned it on the last slide. We use tyrosine epoxy. Here I'm showing an example of where we compared identical devices. So we made the same device from 8 different materials, various materials by micromilling. This includes also industrial polymers such as COC, polycarbonate, polystyrene, polypropylene, Teflon, PMMA, and also, of course, PDMS, which is also produced by molding. And then we simply pipe it into those devices, or we load those devices with 6 compounds with very different physicochemical properties, purely incubate either 2 hours or 24 hours, and afterwards measure how much of those substances is still available biologically in the supernatants. And what we see is really a drastic difference. So in those PDMS devices, PDMS being very notorious for drug absorption, we see that after 24 hours and already after 2 hours, we see that more than 99% of the material of the compound is actually absorbed into the bulk material. So it's not biologically available anymore. Whereas for tyrosine epoxy, we see that absorption is up to 1,500-fold lower. And this is the effect is stronger for hydrophobic compounds with log p values higher than 2.5. So this is an important part that is to be considered, and we then next analyzed whether that could also have effects on pharmacological and toxicological interpretations. And so what we did here is we now incubate our liver spheroid model that I presented briefly previously in either, again, identical devices, either made from PDMS or tyrosine epoxy. And what we see is that chlorpromazine is a hepatotoxic compound. Already morphologically, we get good indications for toxicity in the tyrosine epoxy devices. And then if you compare, again, hydrophobic compounds, chlorpromazine and tamoxifen here, and we compare that between PDMS indicated in the purple bars here and tyrosine epoxy devices, otherwise identical, indicated in the green bars, we see that we pick up the hepatotoxicity of those compounds highly significantly in the tyrosine epoxy devices, but not in PDMS. So meaning that this absorption really masks the toxicity of those hydrophobic compounds. And so now, next step, we integrate them in microfluidic devices. So this is the system that we've established for this purpose. It's a pneumatically actuated device. It works by pumping air into the central chamber. We can then have different tissue models in those satellite chambers here that are connected to the central chamber. And what then happens is once we pump air into the center, this air displaces the medium flows to the side, and then afterwards we release the pressure and the medium very gently flows back. And this setup has a couple of nice advantages. On one side, we have a gas phase on top of each one of those compartments, which means that bubble formation, which is typically can be a big problem in long-term microfluidic applications, is here not disrupting experiments because any bubbles that would be forming are immediately merging with the gas phase on top. And secondly, by using different lengths of the channels that are connecting the central chamber to the satellite chambers, we can directly calculate and determine the perfusion rate in those individual compartments. And this allows us to mimic tissue-specific perfusion rates of different tissue models that we are culturing, and they're purely by design. And we further integrated the system now with a machine vision algorithm. So this means that we can, sorry, this video doesn't play. So this is one example where we are loading blue dye on one side, and we can nicely see the mass transfer very rapidly. So with this synthetic heartbeat, if you wish. And we've integrated this with sampling ports and the machine vision algorithm. And what this allows us now to do is to directly and in real time segment those images and determine the fill height of each one of those compartments. And by this, we can first correct in the long term, and we can sample very small volumes automatically over long periods of time. So we've run this experiment for weeks. 120 microliters is a total volume in the chip. Those can be very, very stable, and we now have the possibility to, in whatever time frame we wish, to sample small amounts, for instance, 2 microliters every hour or every 2 hours, and replace that with fresh new medium. And so this here is one example. Here I'm showing now the long-term application for the emulation of pharmacokinetics. So we are adding here different pulses of the same compound over 14 days. And we can nicely see that in each one of those days, we see a stable overall exposure curve, stable Cmax, stable T1/2, and stable AUC. So this allows us to really do this long-term exposures in an automatic modality. And as one example for the importance of dynamics, here we exposed now our liver spheroids to acetaminophen, a hepatotoxic compound. In one case, we use a pharmacokinetically relevant exposure curve. In the other one, we use a constant one. And importantly, in both cases, it's the same area under the curve. So this means that the liver spheroids are exposed to the same number of molecules of acetaminophen. It's just that those molecules are distributed differently over time. And when we use the constant exposure, also on the chip, we don't see any toxicity. Whereas as soon as we use the dynamic exposure, we see toxicity within the therapeutically realistic overdose time frame of 8 hours. And this can also be used to emulate the toxicokinetics. We've done Olink secretomics, so we can profile the kinetics of biomarker secretion. And we've also combined this with cell painting so that we can directly look for toxicity markers upon toxic exposures. And now last example, we'd like to show from the modeling of hyperglycemia. So here we do a co-culture of pancreas and liver. So those pancreas cells, again, primary human pancreatic islets that we are culturing on there, they still have all the alpha, beta, delta cells. They are responsive to glucose bolus. So once the glucose concentrations reach postprandial glucose levels, we see that they secrete insulin, whereas they don't if they use fasting glucose concentrations. And also this insulin secretion is within the relevant timeframes. And now on the other side, in the liver, we see that the hepatic insulin response genes then are suppressed as soon as we inject the glucose bolus. So this really mimics this axis of communication between pancreas and liver that is characteristic of the regulation after food intake. And we've also, again, done RNA sequencing here to see that triglyceride biosynthesis is up, AKT phosphorylation is up. We've also validated this by Western blot and so on. So this allowed us to reconstruct such an interaction network. Again, I've gone through this very quickly, but we have data for all of those individual steps where glucose triggers insulin production, insulin secretion. We can measure this also dynamically, but in the islets, this goes hand in hand then with an increased ER folding load, unfolded protein response, and ER stress, which is also shown in hyperglycemia. And on the other side, the secreted insulin then triggers or signals via the insulin receptor, triggers AKT phosphorylation, activates branched-chain amino acid catabolism, SRABP activation, and downregulates gluconeogenesis, which then provides a negative feedback for the overall control of glucose levels in such a device. And in the future, where do we expect this to go? So connecting this also with our other tissue models in an integrated setting, then we can automatically inject 3 times per day high glucose concentrations, so postprandial glucose levels. The glucose gets distributed in the device. The pancreas realizes that glucose concentrations are elevated, secretes insulin, and the insulin triggers corresponding glucose uptake in the glucose sink tissues. And for this, we use physiological scaling, which means that we are adding the right amount of these tissue models in order to mimic their physiological functionality. And by then comparing healthy settings versus disease settings, so where we are taking, for instance, material from patients that are obese or that are already insulin resistant, or we take less islets from late-stage pancreatic failure, then we can disrupt this glycemic control, reach the hyperglycemic range, and then ask, okay, which compounds can we now add in order to restore glycemic control? And so in summary, I would hope to have conveyed the message that we've established multiple different organotypic primary culture models, liver, adipose, pancreas, skeletal muscle. I've gone through this quite quickly, in importantly identical chemically defined media composition that allow this crosstalk emulation directly on the chip. These models are extensively characterized. We've by now accumulated over 50 publications and have used them for a variety of compounds, also some that already made it successfully into the clinics. The model portfolio is further complemented by various models of solid tumors. I didn't have time to go through this today in this short time. NanoRim, this fabrication method, constitutes a versatile method for the rapid and scalable fabrication of complex devices without backend processing. We fabricate these devices by NanoRim using tyrosine epoxy systems. These are biocompatible and reduce drug absorption drastically by multiple orders of magnitude. Machine vision can assist this automatic MPS and allow for the emulation of long-term clinically relevant exposure regimens. And lastly, exposure dynamics can have really drastic impacts on the outcomes of preclinical assessments. And with this, I would be done and would be happy to take any questions afterwards in the discussion session. Thank you very much.
And so today I'm Jean Marie Gaffney. I'm from BioIVT, and I'll be talking about our HepatoPack platform. And what is HepatoPack? HepatoPack is a long-term culture model. It is a mature model, and it uses tools from the microfabrication industry in order to create this co-culture system. This model was developed by scientists at MIT, Dr. Sangeeta Bhatia and Dr. Salman Khatani. And what's key to this platform is the micro-patterned architecture. And as I mentioned, we do create this using the microfabrication tools, as this is what allows us to stabilize the hepatic phenotype. You can see here in the center image, there are islands of hepatocytes or domains of hepatocytes surrounded by supporting stromal cells. The image on the bottom left here does show these stained islands of hepatocytes. And here in the top right, you can see these gray bars represent the architecture which we actually use and are able to control using microfabrication technology. And what we're working towards achieving is a 500 micron island diameter, and it has 1200 to 1200 spacing. And so that is something that's reproducible across this model. And one of the key questions, as everyone would know, that researchers working with liver platforms, when they're looking for a long-term culture, they want to see that there is CYP activity, specifically phase 1 and phase 2 over several weeks. Here are representatives of looking at CYP activity over a 4-week period. Initially here in these first few days, you can see that the hepatic activity, looking at this CYP activity, is going up over time. So once the hepatocytes are thawed and plated in the HepatoPack model, that CYP activity is recovered and then stabilized. And what we're looking for, and you can see here on the right-hand side, is suspension culture CYP enzyme activity, which would be our gold standard that we would be working towards. And then HepatoPack, this is the data here in a mature HepatoPack model. And you can see it is performing as well as a suspension culture, if not better over time. Then looking at the applications for which researchers use HepatoPack, initially this was developed for researchers working with small molecules. And so a lot of the early work was done looking at metabolic stability, MetID, and toxicity. And the model is amenable to single dose continuous incubations or repeat dosing regimens, which you would need specifically in a tox application standpoint. In the last number of years, the researchers have really been working in the spaces with RNA therapeutics. So the model is amenable to siRNA knockdown, LNP delivery, siRNA delivery via transfection, and GalNAc conjugation. Also in the liver disease space, such as HBV and malaria. And we've also developed a steatosis model as well with the HepatoPack, which we'll touch on today as well. Just to go back one step just to some of the ADME, as I mentioned before, initially a lot of work was done for small molecules. And this is still one of the key questions that scientists turn to BioIVT for when they don't see turnover in suspension, microsome, or 2D plated hepatocytes. They'll turn to the HepatoPack model. This is an example here for their low turnover compounds. Sanofi had looked at 15 reference compounds, specifically using suspension cultures, monolayer, and HepatoPack, all using the same hepatocyte donor across all 3 models. And you can see there isn't HepatoPack does have an increase in the predicted clearance within 3-fold of 93%, whereas the suspension and monolayer respectively were at 46% and 38%. And this really is because HepatoPack we can culture for a longer period of time and therefore incubate low turnover compounds for a longer period of time. And then just looking at a case study for toxicity, many people are familiar with the phyluridine case. This was in the 1990s, and it was something that in the clinic there were deaths that were observed. If going back to the preclinical work, in the preclinical species, mouse, rat, dog, and monkey, there was no signs of DILI. And so using the hepatopac model, both for human and rat, we are able to recapitulate this in the human model. This was not able to be recapitulated in the human sandwich cultures, though. The data here, with a few of the endpoints, we're looking at ATP, MTT, and urea. And the black lines in all three of these graphs is data from the rat hepatopac. So you can see these here are looking at percent of control. So there's not a lot of change, a little bit of change there. All the colored lines, the green and the purple lines, would be 5 different lots of human hepatocytes that were used in the hepatopac model. And you can see for all of these, they are showing a decrease or a drop in percent of control, which is indicative of toxicity in the model. So the hepatopac was able to recapitulate the greater sensitivity in the human versus the rat hepatopac, looking at the phyluridine toxicity. And then moving on to a little bit more advanced model than the hepatopac would be our hepatimmune model. And this does incorporate the use of Kupffer cells. So this is now our triculture model. And it is focused on immune or inflammation-mediated hepatotoxicity. Therapeutic proteins are often cytokines or cytokine modulators. So we'd want to have those Kupffer cells present. Recently, there's interest in using the model for antibody drugs with the researchers that we are working with. And internally, we've been doing work looking at the addition of PBMCs as well to this model. The panel on the far left here is the hepatopac model using HCI. The middle panel is the hepatimmune model, which contains the Kupffer cells. And these here you can see, are stained by the CD68 for these macrophages. And on the panel on the far right is a close-up of zoomed in on those Kupffer cells. The bottom left graph here will show you via HCI, we were able to quantify the number of Kupffer cells present in the model. You can see over this 2-week period here, there's about a doubling of the Kupffer cells, so they replicate very slowly in culture. And then on the bottom right here is an example of looking at one cytokine here was just looking at IL-6. And when you stimulate these tricultures with the Kupffer cells that are present, you will see a release of IL-6. And then moving forward a little bit to a lot of the interest in the last couple of years from those working in the RNA therapeutic space would be gene silencing, and specifically here doing siRNA knockdown via transfection. To give you a little bit of background, the hepatopac model, the first step is the seeding of the hepatocytes, and then the second major step would be the addition of the stromal cells. If we're doing a transfection, that transfection occurs after the hepatocytes have been seeded. We're using commercially available transfection reagent, RNA Max, for this. We do work with clients when we're sourcing commercially available siRNAs. We also have clients who will source or have custom-made siRNAs targeting their therapeutic targets of interest, and also work with researchers who are designing their own siRNAs as well. And this is just an example on the bottom right of looking out 18 days in culture and assessing that knockdown using mRNA, and also looking at 3A4 activity. And initially, a lot of our work was done, as I mentioned, in the therapeutic target space, working with clients. And so we knew that then we could also apply this potentially to the ADME space. So we have applied this knockdown method specifically for CYP reaction phenotyping when working with low turnover compounds. Specifically, since we're working with the hepatopac model and it is geared towards low turnover compounds, we wanted to look at gene knockdown using siRNA transfection as an alternative to chemical inhibitors. There's a few publications already demonstrating the use of chemical inhibitors in hepatopac for reaction phenotyping. Since it's a long-term culture, it's a little more complicated in terms of maintaining levels of the inhibitor in the culture, so it's more labor intensive, whereas the siRNA knockdown would allow for a single step of that knockdown, and that would be maintained throughout the course of your incubation with your compound of interest. And also the siRNA knockdown method would provide an opportunity for decreasing any off-target effects, increasing the specificity of that inhibition. And also the siRNAs provide an opportunity for where chemical inhibitors are not available. There would be siRNAs that could be available targeting certain UGTs, and such as an example. This here is just an example of a traditional low turnover compound, disopyramide, which we know is metabolized by a 3A4 pathway and has a known FM of 90%. And you can see here on the left, this is an example of using the siRNA method. On the right is an example of using the chemical inhibition method. And you can see in both cases, in your control condition where there's no knockdown or inhibition, you will have depletion of that parent compound. Whereas if you do siRNA knockdown or treat with a chemical inhibitor, you can see that there is no depletion of that parent compound. It's not being depleted. So we were able to show that this gene knockdown method is an alternative to chemical inhibition for calculating the fraction metabolized for low turnover compounds. And then hepatocytes typically are characterized by BioIVT for ASGPR expression. And so we use those hepatocytes that are manufactured by BioIVT for use in the hepatopac model. And one of the test articles that a lot of the researchers are coming to us with are GalNAc conjugated siRNAs. And the ASGPR expression on the hepatocytes does allow for the GalNAc to be taken up by the hepatocytes. And so here, this is an example of looking at 4 different hepatocyte donors in the model and looking over a course of a 2-week period. You can see here via HCI staining that we are able to show the expression of ASGPR over time in hepatopac. So it is robust, and it maintains over a multi-week period. On the flip side, we've just shown that the ASGPR is expressed in the hepatocytes in the model. So we want to see that it's actually taken up by the cells. So in this case, we were working with a Cy3 labeled GalNAc siRNA, which was targeting GAPDH. And here, once again, using HCI, we had the same parameters. We're using 4 different hepatocyte lots in the hepatopac model over a 2-week period. And you can see here that the siRNA was taken up in the hepatopac, and it is quantified here on the right-hand side. And similarly, we did look at the mRNA data for this as well, looking at the knockdown of GAPDH, and it does correlate with the gene expression, it does correlate with the ASGPR expression and also with the uptake via Cy3. And then to touch on our steatosis model as well, and this can be used both in our hepatopac model or the hepatoimmune model, the triculture containing Kupffer cells, and this would be for steatosis and MAFLD modeling. There's 2 different formulations which we have used. One formulation contains free fatty acids, oleate and palmitate. Another formulation contains a high glucose fructose formulation. And looking at these panels here, the panel A is just a control, so no treatment. Panel B would be the addition of free fatty acids. So you can see over time, 2-day, 4-day, and 7-day, they are loading the lipids. And this staining is by Bodipi and has been imaged using high content imaging. The HGF condition here, you can see there is uptake there is uptake loading of the lipids as well. And then the bottom panel D is a combination using both free fatty acids and HGF. This panel here in the middle is an example where we started the lipid loading out to day 4 and then removed the media that actually had the lipid loading agents in there. and to see if we could reverse it. So you can see we are able to reverse that lipid loading there. And this staining has been quantified as well. And in this publication that's referenced here as well, we have also done some characterization using a PCR array of genes, which are typically targeted in MAFLD and steatosis as well. So those would be referenced there. And then just to combine both the sRNA knockdown via transfection and also the lipid loading model, putting those together, one of the key targets which researchers often come to us that they're working with our PNPLA3 would be their gene targets. We do know that the I148 mutation in PNPLA3 shows enhanced lipid loading in culture, and so we wanted to see if this would actually be the case with our cultures. So the top panel here is a wild type donor, and then the panel on the bottom here is a mutant PLA3 genotyped donor. And you can see both of these here are non-treated. This is the free fatty acid condition. So both of these different genotyped hepatocytes loaded with the lipids with the free fatty acid in increasing concentrations. And then here you can see with the increased concentrations, we load more lipids in the mutant donor compared to the wild type donor, which would be expected. And so we were able to show that in the model and quantify that here. And then this here does allow us to look at testing therapeutic modalities for this. So specifically what we did here is we took our sRNA transfection protocol, and we were able to use both the wild type donor hepatocyte lots in HepatoPac and the PNPLA3 with the I148 mutation. We did an sRNA knockdown against the PNPLA3, and we were able to show that here, if you have the wild type donors, if you've targeted the PNPLA3 in the wild type, you're not going to have an effect there on the loading of the lipids. Whereas here on the right-hand side, you can see when you do target that PNPLA3 in the wild type hepatocytes, you will be able to show a decrease in the loading of the lipids there. And then just wanted to mention that you can reach out to myself or reach out to BioIVT to further discuss any research questions that you might have related to the HepatoPac model.
Thanks so much, Jean Marie. So if we could have all the speakers come back on camera for the roundtable. And if any of the audience has questions, please put them in the Q&A now. We don't have any currently, but I think we can start off with one from the registration. So it's for everyone. I'm just going to go around and pick on people. How do you define validation of your model? So I'll start with Ria.
Okay, so in terms of validation, I think it depends on sort of the stage of the regulatory pipeline that you might be targeting. So for us with our model development, we are still more in the early stages. So our validation is going to be more about sort of replicating tissue biology and native tissue architecture and native tissue function. But later on in the stages, when you're getting into validation, that might be more about matching clinical data and making sure the model is actually able to reproduce that data with fidelity in vitro as well, so that then you're able to use it predictively in other readouts as well.
Thanks. Jean Marie?
Yep, so for the HepatoPac model, our validation work has been working with clients' companies and work with reference compounds in order to validate the specific application, which might be of interest to them, and relying on that published data to work towards having data sets that would provide, validation data for the customers to move forward with their, R&D projects there.
Great, thank you. Volker?
Yeah, thank you very much. So Indeed, I can only agree with my previous two speakers. So for us, validation has two dimensions. One is to align with the organ and responses, so molecular phenotype and responses in situ. So we want a liver model, yeah, in vitro to correspond to the liver in vivo, and the other one is really reproducibility across sites, and that comes with the characterization using an extensive set of training compounds. So for instance, for sensitivity and specificity of hepatotoxicity, we've evaluated around 150 compounds. For clearance, we've evaluated around 30.
Great, thank you. Alec?
Yeah, more of the same of what the previous colleagues have sort of mentioned. I think validation requires some sort of acceptance criteria, which is defined by the context of use. So for our platform, maybe it's kinetics or just DILI endpoints. And I think, what you've learned, especially if some of y'all were at the MPS World Summit, is that the next step is from validations qualification, which is really FDA's sort of acceptance of this platform for a specific context of use. And there's a couple programs with the iStand and Entree out there. So hopefully moving forward then with our platform.
Great, thank you. Kalhara?
So for us, we have iPS-derived liver model. So what our approach is, we try to look at each stage to make sure that they follow through all the development stages. So we look for markers at different stages to validate that, and then later structural and morphological analysis with functional confirmation. So that's how we validate.
Great, thank you. Muhil?
Thank you. So in our cases, we are mostly focusing on scalability, reproducibility, and the functionality of liver organ that is developed on our device. We haven't really looked into therapeutic compounds or the model compound validation yet, but we hope to collaborate with other pharma company and then validate our liver organ models in our device. Thank you.
Thanks. Nick?
Right. It really depends actually on whether the model fits for its intended purpose. When it comes to general liver biology, of course, we are trying to see how the different cells mimic liver biology, especially when it comes to in vivo models. Specifically, when we are focusing on disease models, we are interested in the pathophysiology. And what is quite important actually in our field is whether the model is reproducible and robust in generating the same phenotype. And of course, when we talk about DILI, it is about the correct identification of the toxic versus the non-hepatotoxic compounds.
Great, thank you. Sarah?
Thank you. So I think that validation depends in the different criteria. So in our case, first we showed the validation as a technical step. So by showing the reproducibility, experimental reproducibility between different operators. And then when we use our model, actually, our threshold is at the Cmax. So it is a clinically relevant concentration, and also we compare. So our benchmarking is used depending on the FDA-DILI rank. And finally, I think that at the end a good model is a model that reflects real-world reality. And as shown in some of our results, we have case studies where we show the correlation between our in vitro model and clinical outcome in some patients. So yeah, and we are also part of the DILI qualification, so FDA DILI qualification program.
Great, thank you. And finally, Vignesh.
I think just more of the same, but mainly like cross-industry collaborative qualification efforts and really trying to figure out if something goes wrong, why it went wrong, and trying to pass on the knowledge from these kind of large cross-industry collaborative efforts. That also helps a lot as well, I think, as well. And trying to see if there is any parallel effects as well. So if you can qualify liver for one context of use, maybe is there also for food safety also possible? Sometimes the same compounds can apply. That could also be an interesting approach as well.
Great, thank you. And I know we're right at 1 p.m., but we have one question in the Q&A, so if everyone can stay, that'd be great. If not, I totally understand. But we have this question. There are a lot of great liver MPS in the field right now. What does the panel think is missing? So if anyone has a hard opinion on this, go ahead and speak up.
Hi, this is Akisa Sadri from FDA. I just
Yep.
wanted to say that, one of the important markers, I guess, from clinical studies is looking at bilirubin. And that's something that obviously none of the platforms can do yet. I don't know whether any of them are going to take steps to try and, address that, whether you can incorporate, heme-containing protein into your media or whatever, to be able to determine any effect on bilirubin. But if you look at the guidance document on sort of drug-induced liver injury, when we're looking at some of the changes in enzymes, there's like 3 to 5-fold increases, which really we don't see so much in the in vitro system, and also the bilirubin production. So I think that that may be one of the areas where people may have to kind of like focus on in the future, but I don't know. I mean, I'm sure that there's complexities involved in this from a technical perspective, but that was just from a context of use and sort of practicality for regulatory use or purposes. I just wanted to mention that.
Yeah, thanks so much. Nick?
Yeah, going back to Emily's question, I guess this is the $1 million question. There are a lot of things that are still missing in the field, I think. Of course, the first thing that comes to mind is the interdonor variability. We know that we are measuring a lot of Caucasian and Asian, but also there are populations that are not recapitulated in those MPS units. Immune-mediated liver injury, in particular introduction of T cells into the system, we all appreciate the presence of macrophage and Kupffer cells. Another thing that's quite important in the liver field is zonation. Liver is extremely heterogeneous, so being able to generate an MPS that is systematically zonated is something that would be quite informative when it comes to how different drugs are metabolized by what cell types. And of course, I know there's a lot of work going on with different organ interactions, but I think it would be quite nice to have all of them at the same time into an MPS model.
Yeah, thank you. Sarah?
Yeah, just going back to Nakisa's observation regarding bilirubin. So this is really interesting because this is something, maybe I was going really fast during my presentation, but this is something we already measured in our model by comparing actually our in vitro response with the in vivo, the clinical DILI outcome in the patients. And actually we related the bilirubin release with the DILI in those patients. So yeah, this is really interesting to measure bilirubin in the in vitro models.
Yeah, I actually saw that in your slide there was a mention of bilirubin, and I was curious exactly how that happened. So yeah, thank you.
Thank you. Thank you. Anyone else want to talk about what's missing in liver MPS?
I'll second the B and T cell addition, like adaptive immunity.
Yeah.
I think there's some pretty good evidence that like idiosyncratic DILI is due to those sort of mechanisms. So it's big hill to climb though.
Yeah. Thanks. Anyone else? Okay. So I think we can go ahead and wrap up there. Thank you all for presenting. And thank you for the audience for your questions and for sticking around. Really appreciate it. And I hope everyone has a great rest of your day. We can see them.
All right, perfect. Thank you so much. So first of all, hello everyone, and thank you to the 3 Rs Collaborative and the IQ-MPS for the opportunity to present today. My name is Aditya Bhalera from Organo Biotech. And today I'll be sharing our work on developing a human intrahepatic bile duct microphysiological system for mechanistic disease modeling. So this work was led by Brittany Tian, Dr. Shinichiro Ogawa, and Dr. Boyang Zhang and collaborators, and was recently published in Advanced Healthcare Materials. So I'll first introduce Organo Biotech briefly, then I'll discuss the unmet needs in cholangiopathy research. I'll describe our platform and finally highlight how it can be used to study disease mechanisms and fibrosis. All right. So Organo Biotech, a little bit about us. Organo Biotech is a Canadian company based in Hamilton, Ontario. Our technology originated at McMaster University and is supported by multiple patents and peer-reviewed publications. We develop organ-on-chip products and provide research services with ongoing collaborations across academia and industry. And here you can see our product portfolio. But today I'll be talking to you mainly about our angioplate platform. So the angioplate 384-well platform supports 128 perfusible tubular structures per plate, each of them embedded in a hydrogel, fibrin hydrogel between inlet and outlet reservoirs. So gravity-driven rocking induces flow. And we also have an open top design, which allows for tissue retrieval for downstream analysis, as well as imaging, as you see in this video. So our platform has been applied across vascular, respiratory, renal, reproductive, and oncology applications, as you see on the screen. But today we'll be focusing specifically on the bile duct model, which was developed in collaboration with Dr. Ogawa's lab at University Health Network STEMCELL Institute. Dr. Ogawa's lab is part of our Organo network, where we work with academic labs to co-develop models and offer them for industrial commercialization. All right. So cholangiopathies such as primary sclerosing cholangitis and primary biliary cholangitis are complex diseases driven by multifactorial groups. So these include altered bile acid composition, inflammatory signaling, fluid flow, and stromal remodeling. So while each of these factors has been studied independently, understanding how they interact remains a major challenge. So this creates several key mechanistic gaps. For example, which bile acid species actually drives injury? How does inflammation affect barrier integrity? What causes loss of cilia and epithelial polarity? And how does injury progress into fibrosis? So these questions are directly relevant to target identification, candidate selection, and therapeutic development. And to answer them, we need a human cholangiocyte model that allows these signals to be controlled individually and in combination. So to address this challenge, we engineered a fully perfusible human intrahepatic bile duct model using iPSC-derived cholangiocytes within the angioplate platform. We began with using human iPSC-derived hepatoblasts. These cells are seeded into a microfluidic channel where they attach, expand, and differentiate into cholangiocytes. So over 2 to 3 weeks, the cells self-organize into a continuous epithelialized bile duct structure with an open lumen. So the result is a perfusible human bile duct that can be exposed to controlled flow, bile acids, inflammatory mediators, and stromal cells. So this slide shows the structural organization of the tubes that we differentiated. So confocal imaging at day 20 reveals strong CK7 expression, which confirms cholangiocyte identity. We also see primary cilia indicated by alpha tubulin distributed along the luminal surface. So if you look at the two regions of interest that we have selected, ROI1 and ROI2 on the bottom panel, you can see alpha tubulin distributed along the luminal surface where we can see them projecting apically into the lumen. So this architecture matters because apical basal polarity is essential for directional bile acid transport, barrier function, and mechanosensing. And this model produces the structural features in a 3D perfuse context, which is a prerequisite for meaningful functional and disease modeling. So to characterize our model, we first introduced flow to see if this improved our model. So by comparing static to flow conditions, two findings mainly stood out. So the epithelial barrier integrity, as you see in panel 1, measured by fluorescent external leakage, is significantly improved under flow at all time points. So we had day 0, day 8, and day 20 as time points. And primary ciliation, which is on the second panel to the right, nearly doubles from static cultures to those under flow, accompanied by upregulation of ciliation and polarity markers like PKD1, 2, and VANGL1 and 2. So this is consistent with the established role of apical primary cilium as a mechanosensor in intrahepatic cholangiocytes, and it underscores that flow is a biological input driving maturation. So next, we investigated whether stromal support could further improve cholangiocyte function. So when stromal cells, which in this case were irradiated OP9J cells, were incorporated under flow conditions, we observed additional improvements in barrier integrity and cilia formation. So on the left-hand side, the first panel, when compared to flow alone, stromal co-culture produced a 60% reduction in dextran permeability and a 1.7-fold increase in ciliated cells, reaching nearly 75% ciliation, with further upregulation of key maturation and polarity genes. So this suggests that recreating aspects of the biliary microenvironment further enhances maturation and functionality. Beyond the morphology and barrier function, we also wanted to confirm functional activity. For this, we used the Flipper membrane potential assay to assess CFTR-mediated chloride conductance. CFTR is a channel transporter that is implicated in cystic fibrosis. So stimulation with forskolin and CFTR potential VX770 produced almost a 32% increase in activity over controls. And this was also confirmed by glycosylated CFTR protein expression on Western blot when compared to the positive control line CALU3. So together, these results demonstrate that the model is not only structurally representative, but is also functionally active. All right, so this is towards more disease modeling. So this slide summarizes how the platform recapitulates several key biological drivers of cholangiopathy. So for panel 1, we first examined the impact of bile acids. So as bile acids concentrations increased, we observed progressive disruption of epithelial barrier integrity, demonstrating that the model responds to bile acid mediated injury in a physiologically relevant manner. Next, for panel 2, we investigated inflammatory signaling. So among the different cytokines we tested, IFN gamma produced the strongest disease-relevant phenotype, including a significant reduction in primary ciliation. So this is particularly important because ciliary dysfunction has been implicated in causing cholangiopathies. For the next study, which was for us to understand whether these disease drivers work together and not in isolation, we combined bile acids with IFN gamma, and we observed significantly greater barrier disruption than with either stimulus alone. So this suggested a synergistic interaction between inflammatory signaling and bile acid stress. And finally, under panel 4, we incorporated stromal cells to model fibrotic remodeling. So here we asked whether stromal cells could be used to model biliary fibrosis. We compared irradiated non-proliferative stromal cells with active proliferating stromal populations. And we used both murine and human stromal cell systems. We found that proliferative stromal cells induce significantly greater dextran leakage, indicating a loss of barrier integrity. And the accompanying images show extensive stromal expansion in green surrounding the bile duct structure. So this is consistent with fibrotic remodeling. Together, these results demonstrate that the platform recapitulates multiple interacting drivers of cholangiopathy within a human relevant system. So in summary, we developed a perfusible human intrahepatic bile model, a bile duct model using iPSC-derived cholangiocytes on the AngioPlate 384 platform. We showed that physiological flow and stromal support enhanced celiogenesis, barrier integrity, and cholangiocyte maturation. Inflammatory markers like IFN gamma induced a disease-relevant phenotype. Bile acids and IFN gamma acted synergistically to disrupt barrier function, and fibrotic modeling was successfully done using both murine and iPSC-derived mesenchymal cells. So overall, we believe this platform provides a useful tool for mechanistic disease studies and therapeutic development in cholangiopathies. So with that, I would like to thank you for your attention.
Thanks so much, Aditya.
All right, so just really quick, Hesperos was founded by myself and Mike Schuler. Mike Schuler is considered the founder of the body in the chip field about 10 years ago. We've both been working in this area about 30 years. We've won prizes in terms of alternatives to animals, use the SBIR program quite well. We work with folks all over the world. We have a 14,000 square foot facility here in Orlando, Florida, with about 50 people in the company. We're a CRO. We don't sell our systems, but we will offer a full service CRO with analytical and modeling capabilities. And we were the first microphysiological system where only efficacy data was used by Sanofi for successful IND that led to a phase 2 trial, which is now in a phase 3. So what do we do differently than a lot of folks is this idea of clinically relevant functional readouts. I've been working on this for about 30 years. And the idea is you go to the doctor's office, they don't immediately stick a needle in your arm, they'll get fluids, take out fluids, look at biomarkers, right? But how are you walking? How are you talking? Are you making sense? They listen to your heart. You check your reflexes. They might ask you about barrier integrity. What we're really checking on is your muscles contracting okay. Is the electrical activity of your neurons and cardiac cells functioning properly? Is your neuromuscular junction working well? Is the barrier integrity of things like GI tract and kidney functioning? And so basically what I've been doing is taking silicon devices, using surface chemistry to integrate cells with them. So I can take a cardiac cell, and so when it contracts, it bends, deflects the laser, I can get force. I can pattern cardiomyocytes over top of microelectrodes, be able to get conductive velocity and QT interval. I can get force from skeletal muscle. We have a wide variety of different neuronal systems I've only touched on. And then we also have a liver system, which is fully characterized, where we can monitor its activity. And what I'm going to focus on today is to how all these different functional readouts combined with liver gives us a lot of safety and efficacy data. Because we can use these systems non-invasively for acute, but more important, chronic monitor these systems. We publish these out for organ systems in serum-free media out for 28 days. And we're trying to do this without cell death. We can still monitor cell death. What we're really trying to do is to see the deficits in function that would correlate directly with the clinical outcome. So what we can do, we can then culture out liver in addition to other cell types on their own chips, time it so that it be assembled together. We put a gasket on top of it that defines a fluidic channel. So we can then have the serum-free media flow over top of all the different organ systems at the same time. And we've taken these systems past 28 days. We can put an imaging, a clear top, so we can do advanced imaging. So if you have an organoid you'd like to compare to our model systems, we can do that fairly straightforwardly. I just touch on this really quickly. We've been able to supply information for efficacy for a number of diseases that led to phase 2 trials for CMT with Vanda Pharmaceuticals, ALS for Apellis, ALS for Argenx, myasthenia gravis for Dianthus, multifocal motor neuropathy and CIDP for Dianthus, and then CIDP for Sanofi as well. There's many others I can't talk about. So getting back to the multi-organ system, Mike had taken the equations to use to predict the phase 1 dosing, modified those, come up with the first multi-organ system published back in 2004. So more advanced systems here looking at opioid overdose and recovery, and just basically showing that the foundational papers for this go back to 1995 from Mike's lab. We published a 4-organ system with L'Oreal 2016. And again, this is just sort of a layout one of these model systems. Mike had also invented this idea of a gravity-driven system. This was covered in lab on a chip in 2010. And again, using gravity, we get rid of all the external tubes, all external pumps, get rid of the bubbles. It also makes it a low-volume system. So very importantly, and I will emphasize this throughout the presentation, is that we have a liver present. We can see the effect of both parent and metabolite in the same system. So again, we can culture out hepatocytes in 2D and 3D out to 28 days and maintain good viability. We get bionucleotide innovation in the systems. We can maintain CYP activity. We've monitored over 20 CYPs in these systems, and we have this quite well. And these are primary liver cultures. So use basal activity and induced activity. This is published in Biotechnology Progress, again, out to 28 days. Again, these were only hepatocytes. We can add in Kupffer cells and stellate cells as a variable. In this case, we did this looking at imatinib, does not have liver tox, diclofenac does. But what we showed is that the diclofenac was really affecting mostly the stellate cells as opposed to the hepatic cells. We can also induce fibrosis in these systems, other conditions in the liver. In this case, we did it with TGF-beta and CTGF, and do this fairly straightforwardly. Now, what we did is partner with AstraZeneca, where basically we have a heart-liver system where we coupled the heart with the liver in the same recirculating media. They had a compound go through preclinical, and as soon as they went into an animal, they saw a large QT effect. So what we did, we looked at a heart versus a heart-liver system. For terfenadine, terfenadine was an antihistamine that was pulled off the market because it caused long QT. Its metabolite, fexofenadine, now marketed as Allegra, does not. You see heart only, you see a large QT effect, but the liver shifted far to the right. You see with the conduction velocity, you see a no effect with liver present, IC50 at 33 micromolar, okay, with the heart. Anyway, again, we're not killing the cells. What's very important is here, we can also do PK/PD modeling with these systems. We can then grid it up to be able to look at the flow rates, to be able to look at stress. We can look at absorption. All compounds absorb to some degree onto the plastics in these model systems. Like terfenadine absorbs a lot, fexofenadine not so much. We can also then look, model mixing. We can also look at the loss due to absorption, loss due to metabolism, be able to get the air into the curve. With the air into the curve, we can then create a PK/PD relationship. We can look at the PK, okay, of the absorption, PK, and metabolism. Again, since we're measuring QT interval, it's a direct readout that you measure clinically. We can then look at the pharmacodynamics of that. In this case, terfenadine works on the hERG channel, which is inside the cell. We added a barrier that mimicked the cell membrane. We got the true PK or PD. We then extrapolate this to predict guinea pig, dog, and non-human primate data that AstraZeneca had. This is published in Nature Scientific Reports. We've been able to now expand that, where we now have a malaria on a chip model for a digital twin, where we have liver, splenocytes, and endothelial cells. We can actually add blood into the system to show the correct parasitemia, where we go from the ring stage to the to the early Shinsan to the late Shinsan stage, and then what we did is we monitored these 5 different parameters to get the active concentration, be able to predict the PK profile for chloroquine, which is a known antimalarial, and be able to get that, and then we then predict the pharmacodynamic profile for these systems. They be able to predict maximum tolerated dose, no adverse effect level dose, and EC50, which is well in agreement. We then put this into the digital twin and basically show that the acute dosing we did for this MPS in vitro work did not really predict the in vivo outcomes, but now we put it into digital twin. We now have the 4 different dosing schemes that they normally have for chloroquine, and we see we get the correct parasitemia. This was published in Advanced Science late last year. We can also look at both efficacy and off-target tox in this model system. Again, Roche asked us to take the liver, cardiac with 2 different cardiomyocytes, 2 different cancer cell lines, one which was a multi-drug resistant cancer, one was not multi-drug resistant. So for the non-multi-drug resistant cancer, they picked tamoxifen. Tamoxifen by itself is not a good chemotherapeutic. However, if you have a liver present, the metabolite is decreases proliferation. The metabolite by itself decreases proliferation. And then what we also saw is that for the multi-drug resistant cancer, no decrease in proliferation with tamoxifen with liver, and the metabolite we added in a second drug, verapamil, basically showed a decrease in proliferation because it's blocking the chemotherapeutic as fast as it's coming in, it's kicking it out. And we also then showed verapamil is not acting as chemotherapeutic in the system. Again, showing the value of looking a multi-organ system, okay, with the ability of the liver to do metabolism and be able to look at both the metabolite and the parent concentrations. So then we got very interesting. We looked at the off-target toxicity. Tamoxifen, conduction velocity goes down but recovers. Force goes down but recovers. Beat frequency goes down but a little bit recovers. Now for the drug-drug combination, conduction velocity goes down but does not recover. Force goes down but does not recover. However, beat frequency goes down and recovers to some degree. Now, say if you're only looking at beat frequency, you'd say, well, maybe this is toxicity you can work around. But as soon as you put it into an animal for the safety testing, the heart would stop functioning and you'd have to start over because, again, we're not killing the cells in these assays. Again, just showing again, the liver was not affected in this model system. However, it was able to maintain its function to be able to do metabolism. And this is published in Science Translational Medicine with Roche. We also have a CNS model. So I'm going to show how we can couple the CNS to the liver model system to be able to look at both parent and metabolite again. And again, this is a LTP model that is a cartoon I developed a long time ago, but really show we can actually reproduce dual pass clamp electrophysiology, showing we could simulate on these electrodes, record on these electrodes, and be able to reproduce what you would normally have to do with glass micropipettes in an animal slice. We then could then actually measure the signals in these systems and just show we could actually hit it with lidocaine after each experiment to show we get, because MEA noise can be made to look like neuronal signals very easily. We subtract that and be able to get the neuronal aspects. This is published in stem cell reports. But what I wanted to highlight on this is Bayer asked us to create a stress model in this model. In this system, we added in cortisol. We say hyperexcitability, suppression, and excitotoxicity. And we see actually an increase in baseline. And what they wanted to do is to monitor the single organ system. In this case, they want to look at echinacea extract effect on the stress model. We see CBD also had an effect. Now you also then see that after 24 hours, you see that CBD is persistent, echinacea extract is not. Then we had them they had us create a GI tract, liver, BBB, CNS model system. And now you can actually see in the multi-organ system, CBD has a greater effect because it's a prodrug. And now with the liver present, you can see that echinacea extract, we do not see early on. Diazepam as a control, we shouldn't see any effect with that. But you see after 24 hours, we still maintain the activity of CBD. And now we see echinacea extract in this model system. We also worked with Vanda Pharmaceuticals. They had a IND that was being held up because they had a 3-month beagle study. They were being asked by the FDA to do a 9-month beagle study. What we did is a 4-organ system with that model system to be able to look at potential tox for their model with proper controls. That lifted the IND now, and then one of the indications was being held up has now been approved by the FDA for motion sickness. And in brief, this is the model system. Again, a liver to be able to look at metabolism because we had to look at metabolites as well as parent in this model system. We looked at cardiac force, cardiac electroactivity, CNS model, and then also kidney. And what we did is we did a positive tox control for each organ system in this model to be able to produce the data that was submitted to the FDA. We can also look at the results of the innate immune system. We can add in monocytes. We show that the monocytes flow is not being affected by the presence of the cells or the materials, so we can actually get them unactivated. And what we're able to do is to be able to selectively insult them to be able to create the M1 versus M2 phenotype. This is published in Advanced Science with Roche. We're also working on 3 organ comorbidities models, where we're looking at combinations of cardiac, muscle, and liver. We published the sarcopenia muscle. We're just submitting the comments in for review for a sarcopenia cardiomyopathy model. But we're also working on a sarcopenia MAFLD model, a sarcopenia cardiomyopathy MAFLD model. And again, this is our initial FFLD model. We take hepatocytes, pancreatic beta cells, and adipocytes in the same recirculating serum-free media. And we actually then look at a diabetic versus non-diabetic, looking at high glucose, low glucose. We also looked at the effect of inflammation in this model system and showing that the inflammation has a large effect on the accumulation of lipids in the liver in this model system. So I know I went through a lot of information fast, but I really wanted to show you is that what we can do is take a very well-functioning liver system and couple it with all kinds of different other organ models, and we can actually then do PK/PD, of these model systems for both parent and metabolite to predict in vivo outcomes. Thank you much for your attention, and I'll take questions.
Good morning. My name is Jelena Vukasinovic, and I'm pleased to present Lena Biosciences' mechanistic approach to DILI. Drug-induced liver injury is frequently detected late. Preclinical species do not always show DILI. DILI often occurs in a small percent of patients, and certain individuals are more vulnerable than others. However, testing hundreds of individuals in a dish is impractical. This figure shows a common function that fails with acetaminophen and how much those responses vary with different donors. The x-axis is a multiple of Cmax. The y-axis is a cell function normalized to vehicle. The vehicle is the control without a drug. While we need many more donors to estimate population variability in a dish, this may be a good starting point to highlight that there are outliers. DILI is a multifactorial problem, which is difficult to isolate with a single assay. We believe that a battery of mechanistic assays can be used as a prognostic marker for DILI in vitro. The first cell function to fail, the trigger function, tells us what the problem is. If the cell cannot compensate, adapt, or recover, the failure of the trigger function initiates a mechanistic cascade, which may lead to DILI. Now that we define the purpose of mechanistic assays, the question is, how can we account for population variability? First, we propose a 10-donor pool as a practical model of an average human. If an average human means an average metabolizer in the context of small molecules, then we propose a poor metabolizer and a rapid metabolizer to cover the opposing ends of the spectrum. For practicality and cost, we often use the poor metabolizer model, a cell line, as our safety baseline. Here is what the baseline told us for 12 drugs. Note that not all functions were assayed for all drugs on this slide and next. For this and the next slide, we used the poor metabolizer model, a cell line in CDZ scaffold in 96 well plates. This is a simple model without perfusion and synthetic hemoglobin oxygen carrier, which are used in our perfusion well plates. The cells were seeded at 50,000 cells per well. All drugs were administered at a final DMSO concentration of 0.1% and dosed once within 2 hours after seeding. The readouts were taken 24 hours after drug additions. The IC50 over Cmax for the trigger is listed in the figure caption for each drug, along with the Cmax used as a reference. Based on a small number of drugs, our data suggested that a simple model may be able to work, and that a battery of mechanistic assays may be able to differentiate DILI potentials. With that, we decided to run mechanistic assays for both the poor and the average metabolizer for the 3RC DILI project, on top of the standard assay battery requested. Independent analysis of the 3RC DILI study results is ongoing and will be available in future publications. The results presented here are the results of our analysis. Here is what the assays found and how we interpreted our results. Tolcapone was ranked more toxic than entacapone. Our DILI ranking method had 5 levels: maximal, very high, high, moderate, and low. We described in the report what each of those levels actually mean. For entacapone, we noted in the report that the exclusion of poor metabolizers and patients with known liver liabilities should be considered if liver function tests suggest the same. The final rank was based on the cell line, our baseline. The average metabolizer did not raise any flags based on the mechanistic assays. Triglycerol was ranked more toxic than pioglitazone. With pioglitazone, the solubility limit was exceeded for the 2 top doses. This was noted in the report. The image shows the presence of crystals and precipitate in the well without the scaffold and without the cells. Despite that, we did not see any functions falling below 50% or below y equals 0.5 at x equals Cmax. Thus, we assigned it a low DILI rank. The basis for decision-making was the cell line. I forgot to mention that for data shown here and on the following slides, the top row is always the more toxic compound in the pair. The bottom row is always the less toxic compound in the pair. The first graph in each row is the 24-hour readout after 1 drug treatment. The second graph in each row is the 72-hour readout after 1 drug treatment. Lumiracoxib was also ranked more toxic than celecoxib. However, lumiracoxib appeared less toxic than triglycerol or tolcapone, which is why we assigned it a rank of very high, as shown in the box. For celecoxib, the solubility limit was exceeded for the top dose, and this was documented. You can see that these data points are marked inside the red rectangle. Trovafloxacin and levofloxacin came close. However, we noted in the report that levofloxacin had the highest likelihood for the overestimation of toxicity due to low to no metabolism in the liver, which was conjectured based on the assays. Thus, trovafloxacin was ranked more toxic than levofloxacin. We ranked levofloxacin as a moderate DILI risk because we cannot assess indirect effects on the liver in a dish. No LPS stimulation was used in any of our assays for any of the drugs tested. The average metabolizer was a 10-donor pool of primary human hepatocytes comprising 15% non-parenchymal cells. Based on these graphs, tolcapone was again more toxic than entacapone. For entacapone, green rectangle indicates that there were no dose responses operated at 50% or less at or below Cmax for the average metabolizer. Drugs which are more likely to be safe usually do not have dose responses assigned the green rectangle, but this is a commonality rather than a rule, and there are many more factors to consider as indicated in the box on the right. The class, the indication, the number of assays, which assays were chosen for a given battery, the relevance of Cmax are some of the many factors to consider. Using structurally similar DILI positive and DILI negative predicates in the same class can help with interpretation, but it is not the only factor to consider. In any case, the results of our assays should only be considered as one data point in the totality of data available. For the average metabolizer, triglycerol was also more toxic than was pioglitazone. As noted earlier, the solubility limit was exceeded at 3.33 and 10 Cmax. Thus, the likelihood of false positives was high. Yet, the drug still showed nothing assigned the green rectangle for the poor and the average metabolizer, which is why it was assigned a low DILI rank. As with the poor metabolizer, lumiracoxib was more toxic than celecoxib. Because celecoxib solubility limit was exceeded at 10 Cmax, the last data point was not taken into consideration, and celecoxib was assigned a low DILI rank. I forgot to mention that for the cell line, we used 14 mechanistic assays with the readouts at 24 hours. We used 6 mechanistic assays with the readouts at 72 hours. For the primary human hepatocytes, we used 12 assays with readouts on day 1 and 6 assays with the readouts on day 3. While the trigger may be more common in a given class, this is not a rule. It is also generally different for different drugs. Thus, without knowing what the drug classes were, we had to run all assays. Using at least one additional assay set would have been helpful for at least one drug pair, but we felt that this was sufficient. For the average metabolizer, levofloxacin appeared more toxic than was trovafloxacin, but as we indicated in the report, this was most likely due to the absence of metabolism in the liver. Liabilities tend to relate to the place of clearance. If there is no metabolism in the liver, the drug won't clear in the liver. The said efflux transporters must be able to remove it. Based on our dose responses, it seemed unlikely that levofloxacin would go into the bile. Thus, the only remaining burden would be on basolater or blood-facing transporters, and that seemed fine, judging from at least one model. Overall, levofloxacin was assigned a moderate DILI rank, based primarily on the cell line, our baseline. Thank you for listening.
Okay. Hello, everyone, and thank you, 3R Collaborative, for giving me this opportunity to talk here today. So today I'll be talking about the development of human iPSC-derived hepatocytes for drug discovery, translational research, and toxicity testing. I am a senior scientist at bit.bio, and at bit.bio we use deterministic cell programming technology, OptiX, to derive iPSC derived different cell types. I will talk to you about what OptiX is and our hepatocytes in my talk today. To start with a brief introduction of the liver and hepatocytes. So as we all know, liver is the largest organ in our body, and hepatocytes comprises of about 70 to 80% of liver. Being the most abundant cell types within the liver, hepatocytes are actually responsible for most of the liver functions. So some of the functions, as I have listed here, are it is involved in the protein secretion, important proteins within the body like albumin, blood coagulation factors. It is involved in the metabolism of glucose, gluconeogenesis. It is involved in lipid metabolism. It is involved in the detoxification. It is involved in the clearance of harmful compounds, clearance of ammonia, urea. It is involved in the excretion of bile from our body, and it is involved in xenobiotic metabolism through phase 1, 2, and 3 drug metabolism enzymes. So there are different models of hepatocytes, primary hepatocytes are the most commonly used hepatocyte model. They are functionally relevant. They can be used for DILI predictability. However, they pose some challenges, like they are difficult to get, they are difficult to scale, and the most important problem or challenge with primary hepatocytes are lot to lot variability because of the donor variability. There are, of course, other models like primary animal hepatocytes and immortal cell lines like HepRGs and HepG2, but both of them are physiologically irrelevant animal-derived hepatocyte, of course, because of species-specific differences, and the HepRGs and HepG2s are derived from cancer patients, so they are not as physiologically relevant. There are iPSC-derived cells differentiated from directed differentiation. However, the challenge with those cells are they often fail to reach the functional maturity. Hence, at bit.bio, we use the OptiX, which is again deterministic cell programming technology, to generate hepatocytes. The hepatocytes that we generate at bit.bio are physiologically relevant. They are human specific, and they can be used to predict the toxicity of different drugs. They are easily available, cost effective, and we heavily characterize them in terms of functions and their identity. They are highly scalable, and there is lot-to-lot consistency between different batches. The most important being we can culture these cells for up to 3 weeks post-thaw, so it makes their use for the long-term studies really reliable. Before presenting you the data on the characterization of our hepatocytes, I want to introduce you the OptiX technology that we use at bit.bio to generate different cell types briefly. So OptiX, as it stands for, is an optimized inducible overexpression system. It is based on Taton system. So it is split between 2 different genomic safe harbor. In genomic safe harbor 1, we have DOX-inducible element RTTA, and in the second genomic safe harbor, we have different transcription factor of interest under the element that is responsive to the RTTA. So when we induce RTTA by DOX, it in response induces the transcription factors of interest, and it allows the homogeneous control level induction of all the transcription factor within the entire pluripotent stem cell population, thus allowing the deterministic induction of the new cell identity within the cells. Here on the right image here, there is an example of homogeneous expression of GFP reporter on the iPS cells, and this was engineered using the OptiX technology. Now I want to show you some characterization data on our hepatocytes. I want to start with this data here. So on this left corner, you can see the bright field images of our hepatocytes. What I appreciate about this image is that the cells have displayed nice homogeneous cobblestone morphology, and the cells are polygonal with the bright nucleus and very defined border, and within and I have highlighted some binucleated cells with these yellow arrows showing that the cells are maturing. And on this graph here on the right, you can see the expression of albumin, which increases over time in culture, showing that cells are maturing in culture. And the expression of albumin is consistent with the downregulation of alpha-fetoprotein, which is fetal hepatocyte markers. And these two together shows that the cells are losing fetal-like phenotype and acquiring more maturity. And this bottom panel of graph shows the albumin secreted by our hepatocytes as measured by the ELISA. And again, we can see that the albumin secretion increases over time during the culture, which also shows that our cells are further maturing. And in this figure here on the bottom left corner are the ICC images of our hepatocytes stained for the pan-hepatocyte markers, albumin shown here in orange and HNF4 alpha shown here in green. Again, you can see how homogeneously our cells express albumin throughout the culture, and so is the expression of HNF4 alpha. We have characterized our hepatocytes functionally in continuous culture. So these are the few functional assays that we have run into on our hepatocytes. The first image here on the left is a periodic acid skip staining. So with this, we are looking into the glycogen storage capacity of our hepatocytes. So the deep magenta color here are actually staining for the glycogen. So this shows that our hepatocytes can in fact store glycogen within them. Another functional assay that we have run is to see whether our hepatocytes can form network of bile canaliculi or not. So we treated cells with CMFDA, and the cell which was observed by the hepatocytes, as you can see here in the middle panel, in the first figure in this middle panel, the green color. And after 60 minutes, this CMFDA is actually excreted through the network of bile canaliculi, which you can see here, accumulation of CMFDA within this network of bile canaliculi, which shows that our cells, hepatocytes, can in fact form bile canaliculi as well. Another important function of hepatocytes is ammonia clearance, and it does so through the urea cycle. We wanted to understand whether our hepatocytes are capable of clearing ammonia from the media or not. So we added 5 millimolar of ammonium chloride to the culture media, and then we cultured our cells with added ammonium chloride or with our standard media for 48 hours, and then measured ammonium chloride in the supernatant collected after 48 hours. And for the control, we incubated some media with or without ammonium chloride. As you can see here, the amount of ammonia measured on the supernatant is much lower than the amount of ammonia actually present on the media-only control, which shows that our cells can in fact clear ammonia, and as it happens through urea cycle, it points to the presence of active urea cycle on our cell. Next, we wanted to understand whether our cells can be used to model some of the liver diseases or not. In this case, we used MASLD, as it is one of the most common metabolic dysfunction for the liver. So in this figure, you can see the lipitox staining showing the lipid droplets here in red color. When cultured cells in the standard CD lipid concentrations in the media, which was 2% or 20% of CD lipids. Again, the cells were cultured for 48 hours in increased CD lipid concentration. And as you can see here, during the in standard culture conditions, our cells have some accumulation of lipids, but not very high. But when we increase the concentration of CD lipids on the media, then our cells can accumulate most of our cells can accumulate lipids, which shows that in fact we can use our hepatocytes to model early stages of mast cell development. Next, we wanted to explore whether we can use our hepatocytes we wanted to profile our hepatocytes for the drug metabolism profile. Here we have used bulk RNA sequencing. In this heat map on the left, you can see the gene expression plot for cytochrome 450 enzyme, phase 1 metabolism cytochrome 450 enzymes, and we have plotted it for HepG2, fetal liver, and our hepatocytes in the middle panel within the red box here, heparses and primary hepatocytes. As you can see here, our hepatocytes are clustering together with primary hepatocytes and HepG2 and clustering away from HepG2 and heparses and clustering away from fetal liver tissues and HepG2, which shows that our hepatocytes are similar to primary hepatocytes or mature phenotype in terms of their phase 1 metabolism gene signature. And here on the right, we have shown the CYP3A4 activity as measured by the Promega CYP450 glow assays. As you can see here, the enzyme activity also increases over time during culture. We also looked into the phase 2 and phase 3 drug metabolism signature on our hepatocytes, like phase 1 drug metabolism signature. Our hepatocytes again clustered together with primary hepatocytes and heparses for expression of phase 2 and phase 3 metabolism genes as well, and cluster away from fetal liver, which also shows that our cells are shows more mature phenotype rather than fetal-like phenotype in terms of expression of drug metabolism markers for all three phases, phase 1, 2, and 3. We wanted to assess the ability of our hepatocyte to predict hepatotoxicity. So we set up an assay where we culture our cells in 384 well plate, which shows that we can actually use our cells for high throughput screening as well. And then we treated cells with 7 different compounds here with different DILI severity categories, where 1 being the most severe and 5 being no DILI effect at different concentrations for 24 hours. And then we assess the cell viability after 24 hours of treatment. And for control, we use primary hepatocytes. As you can see here, between the two figures here, our primary hepatocyte data on the right and our hepatocytes data on right and primary hepatocyte data on left, the profile of the two cell types is very similar. I will take some examples, like for example, sunitinib, which have high DILI severity, which falls under high DILI severity category. We can see reduction in cell viability with increase in concentration both on our hepatocytes and primary hepatocytes. We can see very similar response of both cell types with the drug. And when we looked at valproic acid, which is known to be have less severe DILI effect in vitro culture, both our hepatocytes and primary hepatocytes do not show any loss in the cell viability. The cells remain viable throughout the study, which shows that both our hepatocytes and primary hepatocytes behave to all these drug tested in very similar manner, showing that in fact we can reliably use our hepatocytes for the hepatotoxicity predictability with different compounds. So with this, I'd like to conclude that with OptiX technology, we have managed to make hepatocytes which have more mature-like phenotype comparable to primary hepatocytes, and they show similarity to primary hepatocytes also in terms of drug predictability and expression of different drug metabolism marker. There is lot consistency. We can generate billions of cells in one production batch, so they are highly scalable and they are readily available, and they can be used for high throughput screening as well. So with this, I thank you for your time today. I don't know if we have any time for questions now. If we do, I'm happy to take questions now or during the roundtable. Thank you.
Thank you very much. I'm delighted to have the opportunity to present today. And today I'll be presenting some of our latest advancements on our platform and some of our recent research highlights. But before I get into that, I will first provide an overview of our MPS model of the liver for those of you who are not aware. So at CN Bio, we have a liver 12 plate, which is currently on the market, and that consists of 12 individual wells, whereby in each well you've got a scaffold, and in each scaffold you've got 301 independent pores where primary human parasites or other liver cells can be seeded, and these cells then form microtissues. Upon formation of these microtissues, these cells are then functional and can be used for various assays. Cell culture media flows up through each of the scaffolds and microtissues along through the well and then into the channel. And this is powered by our PhysioMimics core. And functionality can be shown from a number of different attributes. So here I've got an EM image of biokinetically and tight junctions within the cells after formation of microtissues. And these can also be seen here in the immunofluorescent images on the bottom right-hand side, as well as an immunofluorescent image showing production of albumin within the hepatocytes in these microtissues. Now, this system is compatible with other cell types, but that very much depends on the specific use case. So for the MPK, we'd recommend primary human hepatocytes on their own. If you're wanting to investigate toxicity, such as drug-induced liver injury, that would be a combination of both hepatocytes as well as human Kupffer cells. And for disease modeling applications, such as MASH, that would be a combination of hepatocytes, stellate cells, as well as Kupffer cells. So taking a step back and focusing in on hepatocytes, one of the unique aspects of a system is that fluidic flow, the immediate perfusion through these microtissues, which produces functional and long-lived hepatocytes. And here are some data that demonstrate this. So on the left-hand side, we have the expression of a range of CYP enzymes across multiple primary human hepatocyte donors. Next, we can see albumin production over time and stable over 28 days. We've also got a graph here demonstrating CYP3/4 metabolic activity in comparison to more traditional methods of culturing primary human hepatocytes, so spheroids and in the sandwich culture, whereby within the liver MPS, you can see higher levels of CYP3/4 metabolic activity and sustained for longer. And on the right-hand side is a graph from a publication by a DMPK group at Roche, showing observed clearance in the y-axis and predicted clearance in the x-axis on a range of small molecule compounds demonstrating that predictive hepatic clearance. So that was a brief overview of our liver MPS. So I'll now introduce some of our technological advancements that we've been working on in the past couple of years. So we've now developed a higher throughput version of this liver MPS that enables you to go from the liver 12 plate, which contains 12 individual wells, and we've scaled that down into 48 individual wells per consumable plate. So if we look at some of those characteristics between the two different plate types, on the left-hand side, you've got 12 wells, or we can also refer to them as chips per plate. And if you have 6 plates in parallel, that enables 72 chips per PhysioMimics core. And per well or per chip, you've got 1.4 mil of recoverable media and 600,000 cells which are seeded. And that gives you plenty of information for transcriptomics or proteomics and looking at biomarkers. The liver 48 in comparison, you've got the reduced liver chip size. So 48 chips per plate equates to 288 chips per PhysioMimics core, and that enables you to extract 0.45 mil of recoverable media and 150,000 cells per well. And this is particularly useful for looking at screening or therapeutic compounds, and you're still able to generate and receive a healthy amount of recoverable media and cells for transcriptomics and biomarker analysis. But what's really key, and what we've put a lot of effort into, is to demonstrate that you've got the higher throughput, you've got the scaled down version, but you've still got the same hepatic function. So on top, you can see here in liver 12, an image of our scaffold, which in bright field, and next to it, immunofluorescence imaging showing staining of albumin in green and actin in red. And below it, you can see the comparative image in the liver 48. And we are producing these signature donut-shaped microtissues that you can see in our system. And on the right-hand side is data on the production of both albumin and urea, key proteins produced by primary human hepatocytes at days 4, 6, and 8, in both the liver 12 as well as the liver 48. So I'll now demonstrate and show some of our recent research highlights, which will go into 3 main areas. The first being oligonucleotide uptake and efficacy, the second, immune-mediated drug-induced liver injury, and then finally, insulin sensitivity and the importance of cell qualification. So I'll start with our Physiomimics oligonucleotide uptake assay. So this assay can be used to investigate both the uptake of oligonucleotides, which include both siRNAs as well as ASOs, as well as investigating the extent at which the target gene is being knocked down. So in a typical experiment, once the hepatocytes have been seeded, it takes 4 days for those microtissues to form, as I've just shown you. And at day 4, you would then begin to add in that oligonucleotide as either a single dose or a repeat dose every couple of days. And that experiment would go on for typically a couple of weeks. And the types of readouts you can receive are liver functionality, gene knockdown, and uptake of modality in terms of its fluorescence biomarker. And in this slide is an overview of one such study that we performed, whereby we investigated the dose-dependent knockdown of GAPDH within a GalNAc conjugated siRNA. And in this study, we had 3 types of molecules. The GalNAc conjugated to the siRNA for GAPDH, the target gene. The naked conjugated to the siRNA for GAPDH, whereby you don't have that GalNAc molecule which targets a specific receptor in the primary human hepatocytes. And then finally, the GalNAc targeting the scrambled siRNA, whereby you've got that GalNAc molecule, but it's not targeting this particular gene. And this was performed at 3 concentrations, 10, 50, and 100 nanomolar. You've also got a vehicle control here without any of the treatment. And on the top, we've got some liver functionality markers. So you've got cell health in LDH and then albumin, whereby we see essentially no difference in either the compound being added or the concentration at which this is added. Underneath, we have an overview of the uptake of that siRNA using the labeled siRNA in magenta. On top, you've got the GalNAc conjugated to GAPDH, whereby you see an increasing amounts of signal as concentration increases, which is really nice to see. And you've also got something similar in the naked conjugated to GAPDH. And interestingly, there doesn't seem to be too much of a difference at the highest concentration between the GalNAc and the naked when you combine it to target that target gene. On the right-hand side is the gene knockdown, and that's when you can essentially better understand the sensitivities of your therapeutic. And here you can see the knockdown of the GalNAc to GAPDH, as well as the naked and the scrambled. And what's clear is with that conjugation of the GalNAc with your GAPDH, that's where you see the most proficient knockdown. And that's exactly what we'd expect to see here. And this assay is currently being used by multiple customers and clients for lead candidate selection of their therapeutic siRNAs and ASOs. And what's really important is because we're using both human and functional cells, these systems are more translational than if you went to do some of these screens in an animal, such as a non-human primate. Next, I'll pivot to discussing the latest work on our liver immune MPS. We have developed a protocol to enable the circulation of PBMCs, or peripheral blood mononuclear cells. Importantly, this enables the modeling of immune-activated drug-induced liver injury in response to therapeutics such as biologics, which include antibodies. And this data just shows you an overview of some of the work that we've done. So what's beneficial in CN Biosystem is that you can have these recirculating cells in the system, which makes it a really powerful system for modeling immune cells such as PBMCs and studying immune-mediated toxicity. So here you've got PBMC recovery over a range of different PBMC cell populations, as well as the cell viability, which is at or just below 80% for all the cell populations added. On the right-hand side, there's some biomarker data, so albumin, a key functional marker of primary human hepatocytes, whereby you can see here when you've got hepatocytes on their own or hepatocytes with the PBMCs, you essentially get either stable or increasing levels of albumin over the 48-hour time period of this particular experiment. However, when you've got PBMCs that are activated with PHA or PMA, you see that decrease in albumin production. On the bottom right-hand side is a graph of ALT, whereby we see essentially no change in ALT in each of these different conditions. Next, I've got an example using our drug-induced liver injury immune assay with the known DILI risk antibody ipilimumab. Now, when we conducted this particular experiment, we were keen to measure the concentration of some inflammatory cytokines to understand what the response was when we added the addition of the antibody. And what's really clear from this study is that you can see an increasing level of IL-2 upon the addition of the antibody with the hepatocytes, but importantly, we particularly see this response when you've got the activated PBMCs. And this is true for both IL-2 as well as for TNF-alpha. When we look at LDH, we see a separation in the data. So that separation occurs between the PBMCs that are activated and the PBMCs that are not activated, or when you don't have PBMCs in the system. For albumin, the data is quite stark, whereby at the end of the experiment at day 8, you see a really interesting and expected separation, whereby albumin essentially goes to 0 when you have that PBMCs which are activated alongside the primary human hepatocytes and the monoclonal antibody. So to summarize on this part, recirculation of immune cells enables the study of immune-mediated DILI, and we can demonstrate sensitive detection of liver responses, identifying early changes in key liver function markers. And we're undergoing further work using more antibodies, so watch this space for more data to come. And finally, I just want to end with a collaboration that we have been working on with Novo Nordisk for the further development of CN Bio's MASH model using a liver MPS. As part of this, rigorous cell qualification was really important, and it's a requirement when undertaking disease modeling. And what was important in this study for Novo was identifying insulin-sensitive primary human hepatocyte donors. So in this study, 23 donors were tested from multiple cell vendors, and insulin-sensitive donors were a key requirement. First, this work was first carried out in 2D using primary human hepatocytes. And this data shows a subset of that insulin-sensitive work. And you can see this work was carried out at increasing concentrations of insulin and then measuring the glucose output. And in this work, 6 out of the 8 donors exhibited some level of glucose output inhibition in response to insulin. But one of the key questions was, after first conducting this in 2D, was, does insulin sensitivity in 3D correspond to results in 2D? And when I talk about 3D, I'm referring to the liver MPS, whereby we have the formation of the microtissues, which are in 3D. So we performed this work, and 11 out of the 23 donors were sensitive to insulin in the liver MPS. And you can see insulin sensitivity really clearly, whereby you first measure glucose at 0 nanomolar insulin, and then at a concentration here at 3 nanomolar. And if you've got insulin sensitivity, you get that reduction in glucose measured. And what was interesting was that that 2D did not always translate to 3D insulin-sensitive inhibition of glucose output. And you can see this in the green stars for these particular donors, shown here, whereby you have really nice insulin sensitivity in these 3 hepatocyte donors. In the red cross, we see insulin sensitivity in the liver MPS or 3D, but we did not see that in the 2D studies. So to summarize here, self-qualification is important, particularly for disease models, and we were able to demonstrate that within the liver MPS, you can culture insulin-sensitive primary human hepatocytes, and that can be used to screen these cells. So to summarize, I've shown data on the CNBio liver MPS system and shown that long-lived, highly functional hepatocytes are supported by the physiologically relevant flow through the microtissues, as shown on the right-hand side. We have now on the market a liver 48 plate, which enables you to work with liver MPS in a higher throughput fashion, particularly beneficial for screening of therapeutic compounds. I've also demonstrated broad applicability across research areas that includes oligonucleotide uptake and efficacy, immune-mediated DILI, as well as disease modeling, including MASH. And finally, I've shown you some data on the importance of robust self-qualification to ensure reproducibility and confidence in experimental outcomes. Thank you very much.
Thanks so much, Yasin.
So thank you to the 3RC organized first for the opportunity to present Xellar Biosystems Liver MPS model today. So I'm going to talk about how Xellar organo-chip technology is transforming a non-clinical evaluation by connecting scientific innovation, industry needs, and evolving regulatory expectation to improve the drug development and safety assessment. Xellar Biosystems, founded in 2021 and located in Boston, USA, and our organo technology integrates high-throughput screening platforms with advanced image-based AI to support end users' predictive drug discovery and development. So we also provide both service and product. So with the global phase out of animal testing and the pharmaceutical and beauty industry are witnessing a major paradigm shift from the conventional assays to NAMs. So traditional animal testing faces significant ethical challenges and often suffers from lower predictive accuracy and lengthy development cycles. By leveraging organo-chip and solid organoid technology, we can achieve higher predictive power and then significantly accelerate the cycles and fully align with the global 3R principles. This slide highlights Xellar's vision for scaling organo-chip technology through automation, data integration, and AI. Starting in the upper left, we are developing a large format device that maintains the same chip level design while increasing throughput. In the upper right, web automation enables automation of the entire workflow from chip handling to dosing and sample collection. This reduces the human error, improves the reproducibility, and significantly increases operational efficiency. In the lower left, we leverage AI-powered analysis to extract a deeper insight from the complex biological data. AI can support image analysis based on the bright field image, phenotype classification, target discovery, and toxicity prediction, and accelerate the decision-making and reduce the manual analysis time. Finally, in the lower right, the data integration brings everything together through our own cloud-based data science platform. So together, these capabilities create a scalable, data-driven ecosystem that enables more efficient and predictive organ-chip research and development. So this slide showcases Xellar's OCPlex platform, which is designed to provide a cost-effective, scalable, and flexible solution for organ-on-chip application. On the left is a chip cross-section view where we combine the microfeature engineering with the surface coating technology to create precise hydrogel patterning. The platform is available in both 2-channel and 3-channel configurations, allowing researchers to select the level of biological complexity that best fit their application. For the 2-channel format, we offer OCPlex A3296 devices, where the 3-channel format includes OCPlex 4664 devices. This range provides flexibility from low throughput assay development to higher throughput screening studies. This slide highlights how the OCPlex platform provides purpose throughput and biological complexity. On the left, our platform supports both, as I said, the 2-channel and 3-channel disease models. The 2-channel design enables interaction between the vascular and tissue compartments, making it suitable for many disease and drug testing applications. The 3-channel configuration adds an additional tissue compartment, allowing more complex models that better recapitulate in vivo physiology. On the right, we offer 2 flow control options. The gravity-driven system uses a locker platform to generate a bidirectional flow, providing simple and scalable solution for the higher throughput studies. For the pump-driven system, enables a single-direction flow or media circulation through a quick connect design. These options allow research to select the appropriate balance of the throughput, physiological complex, and the experimental control for their specific application. So these slides demonstrate that our liver chip successfully recapitulate the key human liver functions under both static and dynamic cultural conditions. We evaluated 2 important biomarkers of the liver function, albumin production on the left and the urea secretion on the right, using primary human hepatocytes from multiple donors. For the albumin, all 3 donors maintained robust production over 14 days with dynamic flow conditions generally supporting the sustained liver-specific function. Similarly, urea production remained stable throughout the current period, demonstrating preserved metabolic activity. Importantly, both albumin and urea level exceeded the MPS Consortium benchmark criteria across all donors and time points, indicating that the platform consistently maintains physiologically relevant liver function. This slide demonstrates liver chip's ability to detect the DILI using primary human hepatocytes from 3 different donors. We evaluate 2 well-known hepatotoxic compounds, acetaminophen and triglycerin, across a range of concentration. On the left, we monitor the 3 key endpoints: albumin production, ALT release, and cell viability using live data staining. As drug concentration increased, we observed a dose-dependent decrease in albumin viability along with the increased ALT release, which are classic indicator of liver injury. Importantly, the response varied among the donors, highlighting the biological variability seen in human population and demonstrating the platform's ability to capture the donor-specific sensitivity to toxic compound. This slide demonstrates the predictive validity of our liver chip using 7 days treatment of APOP-induced liver injury in humans. Following the APOP treatment, we observed a significant increase in ALT release, albumin and urea production was substantially reduced. We also observed a decrease in ATP levels, overall cell viability, confirming the loss of cellular health. Importantly, mitochondrial dysfunction was severely impaired, as shown by the reduced mitotracker and TMRM signals, indicating the mitochondrial damage and loss of membrane potential. And following the travoresidone treatment, we also observed significant increase in ALT, albumin, ADP level were significantly reduced. TissUse also caused a substantial decrease in cell viability and both might trigger TML signals. Overall, these results demonstrate that the Xellar liver chip captures clinically relevant TGG and APOP-induced hepatotoxicity and supports its application for the predictive DILI assessment. This slide highlights the liver chip's ability to maintain the drug metabolizing capacity, which is essential for the evaluating drug efficacy and toxicity. On the left, we measure the expression of key phase 1 cytochrome P450 enzymes over 14-day culture period. Notably, CYP3A4 and CYP2B6 expression increased over time. On the right, we evaluate the CYP3A4 activity, one of the most important enzymes involved in the drug metabolism. And following the treatment with the two hepatotoxic compound APOP travoresidone, CYP3A4 activity was significantly reduced compared with the vehicle control. This slide compares MRP2 expression in conventional 2D primary human hepatocyte culture with our 3D liver chip models. As you know, the MRP2 is a key biochemical transporter involved in drug and metabolite efflux. So as shown in the left, 2D cultured hepatocytes express MRP2 and also both 2-channel, 3-channel liver chip model. We also observe robust MRP2 expression together with enhanced 3D tissue organization spheroid formation. This slide demonstrates the reproducibility of the Xellar DILI liver chip using APOP hepatotoxicant. So on the left, we evaluate the APOP response across 9 independent experiment runs. In every study, APOP consistently reduced albumin level by approximately 7 to 80%, demonstrating robust and reproducible detection of liver injury over time. On the right, we assess the reproducibility across 2 independent sites using a panel of the 10 compounds with known high or low DILI risk compound. Importantly, the results show the 100% consistent between the sites. This case study highlights a blinded collaboration with an industry partner to evaluate the predictive capability of our liver chip platform for liver toxicity assessment. The study includes 3 undisclosed candidate compounds and an additional negative control tested concentration ranging from the 0.1 to 300 times the human Cmax. The heat map summarized that combined the toxicity scores based on the 4 key readout albumin, ALT, viability, and ATP, where higher scores shown in red indicate a stronger toxic response. As expected, travoresidone is our positive control, showed a strong toxic signal, and the research predict compound 2 and compound 3 as having a toxic profile comparable to the positive control, while compound 1 and showed the minimal effects similar to the negative control. These findings were later confirmed by the sponsor and then supported their go, no-go decision-making process, demonstrating the value of Xellar liver chip for prospective DILI risk assessment in drug development. This second industry case study was conducted as a blinded evaluation with the AI drug discovery and pharmaceutical partner. So the study includes 4 compounds targeting the same biological pathway with known but undisclosed clinical outcomes. Background of the compound is that they tested 2 internal early-stage compounds for potential liver toxicity, along with the 2 external controls. One failed in phase 1 due to the liver toxicity, and the other one was phase 2 compound with no liver safety issues. The goal was to assess whether the liver chip could differentiate compounds with and without liver toxicity and capture donor-specific response. The heat map summarizes the toxicity scores generated from albumin ALT BioIVT measurement using two different primary human hepatocyte donor. As shown that higher score indicated in red corresponds to strong toxic response. So importantly, the liver chip clearly distinguished compound with higher toxicity risk from those with lower risk. We also observed difference between the donors, highlighting the platform stability to capture donor-to-donor variation. This case study highlights our collaboration with L'Oreal to advance annual free safety assessment using human-relevant organochip technology. So as shown here, traditional clinical model typically achieve the predictive accuracy approximately 56%, but the Xellar liver chip demonstrate 92.3% predictive accuracy. This case study highlights our participation in the International Consortium and Broad Institute to advance omics approach for liver toxicity assessment. Xellar is currently participating in this project on an advanced liver model with liver chip. So this slide presents preliminary result from OASIS Xellar DILI project. And then on the right, Pierce correlation heat map compares morphology profile across wells. So we observed modest clustering among wells treated at the same concentration across different plates, indicating reproducible phenotyping signatures. So this study focused on the MPS, and then Nakisa presented yesterday, and we are very honored to be one of the participating MPS company on this project. And this study is summarize our platform's preliminary FDA project, and then this finding and conclusion have not been formally disseminated by the USA FDA and should not be considered to represent any agency determination or policy. So on the right, we ranked the analog pairs using a treatment to mean toxicity ratio. So these results, once structural analog were known, each compound was identified to be either higher or lower DILI within the pair. So, and then this with the collaboration, our Xellar organ-chip LOI also accept in the FDA standard program. Yeah, so we have different more than 10 tissue disease model established. And thank you so much.
Thanks, Jihai.
Hey, my name is Daniel. I'm from Moore. We're a small company in Germany, and we're a little bit different here. We do lab automation specifically for 3D cell culture, but what we have in mind is helping with the quality of like NAM data for example, 3D liver culture. So, like I said, we're a bit of a black sheep here at 3Rs RC. We don't actually engineer any of our own MPS, but instead we want to make yours better. And so first we're starting off with a lot of like 96 well format with spheroids and organoids, but we're also interested in other automatable MPS systems as well. So in general, the protocols we've been working with follow this workflow, phase 1, where we generate the cultures, followed by a quality control and treatment. And treatment here can also be just medium exchange. And between phase 2 and 3, we can iterate on this to keep track of how the culture is maturing, for example, and at some point doing an endpoint analysis. And we strive to make this as end-to-end as possible. So here on the left is our Mobot, and it's essentially based on a liquid handling system, but it's specific for 3D cell culture and has optimizations and features for that. So all the essential functionality is provided by these specific modules. So for example, there is no integrated incubator, nor do we have plans for that. But for example, we do have a heating plate, a warm plate that can maintain warmth of the plates. Also a cooler, so for example, for using Matrigel. And additional features like an on-deck imaging system, which I'll talk about, and also other equipment like a shaker, tilter, which, for example, some SBS format chips would benefit from. We also focus on the software side of things. So here is an example for the liver spheroid pilot hepatotoxicity we worked with for a DILI. Quite straightforward here, but it's something that is like an ELN protocol design. So no coding necessary. It's meant for use by the bench side scientists to plan out the individual steps, for example, here including imaging at a high frequency. And it's very easy to use. Just set up the plates as you want, as if you're doing pipetting yourself in an experimental plan. We also have this modularity on the work field so that you can plan experiments also with exactly how you would set up in the hood. You would typically be working with the plates, the tips, and all of the reagents you need. And this is all for usability. So as a proof of concept, we don't do just liver, but as a proof of concept, we have been using liver spheroids essentially as our first cultural system because of its establishment and also its high demand. So the culture, the protocol we had was over 5 days where we seeded, allowed them to aggregate, then did a medium exchange with acetaminophen as the drug, and then did endpoint characterization. And we did comparisons between manual handling and using the automation system, the Mobot, and also, comparing the untreated and treated. And we see that there is essentially the results are either as good as using manual or in some cases improved, which I'll show next slide. But here we can see that when it came to the treatment during our DILI experiment, that between manual and the Mobot, we had very similar performance. So we can automate these protocols with confidence. So we gain all of the features of automation, for example, like throughput and having the data provenance and confidence in the execution as it was written, and also some potential improvements, as we see here with more uniform liver spheroid generation. So with our imaging system, we can image conveniently as often as we'd like. So we based these data here based on imaging and morphology quantification. We see that with area and roundness before and after medium exchange, there's essentially very little or no difference. So even with careful human pipetting compared to using a robot, we saw no detriment. And in some cases, we see an improvement. For example, if there's any mistakes made by the human doing the manual pipetting, and in some cases, a much better coefficient of variation. As you can see in the roundness, we have a much tighter distribution of the morphology. And we see this here as well over multiple replicates in the area. We see a much improved coefficient of variation. And we believe this has to do with just a more consistent execution of the protocols. And this is already using just the baseline unoptimized mechanics. So we can control all of the pipetting parameters, such as the speed, which in some cases can be causing more stress for the tissue. So here we see that with the automated protocol, with the Mobot, we actually had improved viability compared to manual. And we suspect that is due to how gentle we can be with the handling. And at the very least, it's no worse in some of the functionality, for example, with albumin secretion. So we also allow monitoring of 3D culture using a imaging system that is on deck and in workflow. So as I showed previously, we can image before and after each medium exchange and just have a really high density, temporal density of sampling the features. So we can also feed this back in. And the quantification is done in real time, so you don't have to wait until going back to a workstation, do the analysis, process it, but you get that feedback right away. And so here, for example, when we did a titration of seeding density of liver spheroids, you can see here the heat map showing from darker is larger sizes and area. So on the left, middle, and right, we had low, medium, and high seeding density. And this is the kind of feedback you can get if you have outliers, for example, as an example for quality control. And we're expanding to use other technologies from other companies as well. So here, for example, we're using the Acura plates from InSphero, also culturing liver. And we're able to maintain performance in the imaging and automatic segmentation and morphology analysis and these sort of wells. And also, for example, from Corning, these aplasia plates with microcavities or with any other plates that are SBS compatible, having these sort of features. And so in the future, so we're just starting. So that was the preliminary data we had using HepG2. But now we're currently this summer, starting this week, expanding to more relevant systems, protocols, and also for throughput. So for example, using primary human hepatocytes and spheroids from that, organoids that are iPSC derived from liver, doing a larger DILI screening on both of these, so with multiple compounds. And then also the matrigel context. So this is actually quite a challenge for automation because of the manual techniques required, but we have also developed mechanical solutions to that. Like I said earlier, optimizing the mechanics, the plate layouts to further improve these protocols. And additional high-frequency imaging and doing predictive quality control so that as there is, for example, differentiation of the organoids, we can track and predict if we're able to obtain a high-quality yield before we need to waste resources. And we are also working with additional tissue as well for other drug-induced studies, but liver is definitely one of our interests. So this is our team. We're quite small, but we have a combination of scientists and also engineers, software and hardware, all working together to execute on this. So from made by scientists for scientists, essentially. So we won a new product award last year at SLAS, and quite proud of that. And we're hoping that, yeah, we can collaborate with others and improve the MPS. So if you have any questions, I'll be happy to answer them.
So thank you. It's a pleasure to be here today, and I'm very happy to be given the opportunity to present the InSphero's liver microtissue platform and how it can be used and applied at the drug discovery and development process to assess liver safety and also to do mechanistic toxicology investigations. The 3D inside liver microtissue is a long-lived and physiological model. It is composed by primary hepatocytes of a 10-donor pool of 5 male and 5 females. And these primary hepatocytes are co-cultured together with primary non-parenchymal cells of a crude fraction. Our liver microtissues are self-aggregating in the well, and there are no exogenous matrices needed for this. They are also cultured in a chemically defined cell culture medium with no added serum. And this avoids the batch-to-batch serum variability, which can be seen, and provides a reproducible experimental setup. In addition to our human donors, we also have species-specific liver microtissues. The animal models which we offer at the moment is rat dog, mouse, and non-human primates, and they are all generated by species-specific donor cells. And beyond the healthy donor liver microtissue, we also have the ability to generate customized models based on different demography, genetical background, or also disease models. One important feature of in vitro liver microtissue is its maintained liver functions. We have maintained phase 1, phase 2, phase 3 metabolism, and we also know that our liver microtissues are able to both synthesize and secrete albumin and bile acids.
In addition to this, our liver microtissues are responsive to external stimuli and will then secrete cytokines. The most important part here is the longevity and that the functionality is kept over this culture period. And this is something I would like to go more in detail into in the next couple of slides. Here, I would like to share some data from our cell lot validation and model characterization. And the first parameters we're looking at is to have a stable viability and a stable form and size of the liver microtissue over time. This we assessed by looking at the ATP, basal level of ATP over 4 weeks, and as well as the diameter of the liver microtissue. Both of these parameters remain stable over the culture period of 28 days. In addition to this, it's important to also assess the hepatocyte functionality. This can be done by looking at albumin as production and secretion. The albumin was assessed over 28 days, and we also here see that it remains active and stable over this culture period. As I mentioned on previous slide, our liver microtissues are able to synthesize and secrete bile acids, and we also wanted to look at the formation of bile canaliculi. This can be done by adding the bile acid analog called CLF. And as you can see in the green image here, you have the wire-like networks, which symbolizes or is then bile acid bile canaliculi networked. And that demonstrates that our liver microtissue has forming bile canaliculi and that they are also functional. Moreover, the metabolic activity is very important when you do drug assessment. We here assess the CYP450 activity. We added the specific enzyme substrates into the culture, and then we assessed the formation of its metabolite. This also was done over 28 days. And you can see that we have a stable activity of the different CYP enzymes over the entire culture period. Another important feature of a liver microtissue or an in vitro model is its ability to respond to immune stimuli. This we assessed by stimulating the liver microtissue with LPS and then measured the secretion of interleukin-6. As you can see in the graph on the right-hand side, we have a robust response of IL-6 secretion, and it remains stable for 28 days in culture. This altogether demonstrates that we have a stable viability. We also have stable metabolism and immune competence up to 28 days. And this allows the liver microtissues to be applied for long-term and repeat those exposure studies. Our liver microtissues are aggregated within InSphero's own Acura plates, which exist in 384 and 96 plate format. These plates have a specific well design where the liver microtissue is placed in a secure cavity in the bottom of the well, and it also contains a secure ledge which allows for full medium exchange and minimizes the loss of spheroids during this process. Our plates are also automation friendly, and this allows liquid handler and also then enables larger scale studies. We produce our human liver microtissue weekly, and we have every 3 weeks animal liver microtissue production. Our production processes are streamlined and standardized, and this then provides us with very consistent data. In the graph on the left-hand side, we have collected the ATP content and the diameter of the liver microtissue for different productions over 5 years. And as you can see, these two parameters both remain remarkably stable during this period of time, independent on cell lot changes, different productions, and different operators. The high-density plates, which we are working with, also allow us to include several different concentrations and replicates. In our standard setup, we measure 7-point dose concentrations and in 4 replicates. This allows us to calculate a precise IC50 value and also gives us a more robust measurement. So all this together with the standardized production, the robust measurements, and the cell lot validation gives us very stable and reproducible data. And this is exactly what the graph on the right-hand side is representing or illustrating. Here we have collected the IC50 values of chlorpromazine, which is our reference compound. And you can see that over 45 different experimental runs, independent on productions, cell lot changes, or operators, the IC50 value remains very stable. So our liver microtissues are very versatile, and they have been adopted by both academic and industry researchers. And this can be seen by an increasing number of peer-reviewed publications. We have different studies in different areas, such as species-specific translational research. We have DILI prediction. We can see publications over environmental toxicology and mechanistic toxicology. And this increasing body of publication is also then reflecting the versatility of our model, that it can be used in different research setup. And if anyone would be interested in reading more about this publication, you can find the complete list of them on our webpage. So with this versatile model, I would like to demonstrate also today how it can be applied during the different stages of the drug discovery and development process. And therefore, I would like to focus the next couple of minutes on the different contexts of use cases. So in the early stage of the discovery, during hit identification or hit to lead optimization, we can rank order the drug candidates based on their hepatotoxicity. This can be done by using the liver microtissue and assess their ATP IC50 value, and thereafter rank order your candidate drugs based on the compound which has the best safety profile. And also early on, therefore, eliminate the drugs which shows the less favorable safety profile. As a second context of use, as we progress a little bit further on this profile, we can work with the lead candidates if the human Cmax exposure data is available. We can further assess this by calculating the so-called C2C score, which is the cytotoxicity to Cmax ratio. And by including the exposure data, we can then assess a more relevant value for the prediction of the hepatotoxicity. And we have seen in this publication of Fass et al. From 2024 that we have a high sensitivity for correctly identify the withdrawn drugs. For these two contexts of use, we offer a list of different standardized cytotoxicity assays or screening assays, and all these assays can be performed in human as well as in our species-specific microtissues, and they can be also assessed up to 28 days of culture. The ATP assay can also favorably be multiplexed with different biomarkers within the cell culture supernatant to provide deeper insight into the mechanism beyond the general cytotoxicity. Next, as we progress into lead optimization, it might also be interesting to identify the adverse effect of the lead compound, and this can be done by applying different specific mechanistic assays or by using a liver microtissues to generate a transcriptomic toxicity profile based on the drug treatment. Also here we have a portfolio of different standardized assays which can be applied. Transcriptomic sequencing is an ideal solution to identify toxic reaction signals. We also have different specific mechanistic assays, but due to time restrictions, I cannot go into all these specific assays. However, what I would like you to notice is that most of these assays are available for both human and species-specific liver microtissues. Later in the preclinical studies, sometimes a severe event or an indication can also arise in the preclinical animals. And here it's the next use case comes in, which is to clarify the discordant species toxicity and for the translation to human. And it is difficult when you have an indication in one of the animal to know if this will translate into human. And there are human and species-specific liver microtissues can become a very useful tool to decipher the difference in toxicity profiles. For example, the compound profile in the different species can be compared on transcriptional level, and this can help to not only uncover the mechanism of toxicity, but also to see if this is an isolated event in one species or if it is something which is transferred across different species also into human. The fifth context of use during clinical trials is to evaluate DILI potential in patient population. So it's not always the case that the healthy safety assessment can be applied to different patient populations. The safety profiles might differ depending on genetic background, metabolic profiles, or also co-treatment with other medications. And here we can apply our customized liver models where we can select specific donors with this genetic background, or also apply our our disease model, for example, our mesh model, to evaluate the safety concerns in a specific population. Finally, the context of use is in clinical trials. It could be the case that we have an unexpected indication coming up at a later stage in the clinical trial, and to investigate the liver microtissue becomes a very good closed experimental setup where you can apply different mechanistic assays to investigate the behind reason or mechanism of this indication, and this can be then useful for decision making and also for mitigation strategies. So with this, I hope that I today managed to give you an overview about the liver microtissue, that you can see that it's more than just a screening tool at an early stage of the discovery process. I also have demonstrated that the reproducibility and the stability and robustness, scalability of the model provides it with good features which can be applied at different stages of the discovery process. And with this, I hope that there are some questions, and I would be happy to answer them.
Thanks so much, Anna. So we'll go ahead and get the roundtable started. So if everyone could come back on camera, but mute your mics unless you're answering a question. We already have a couple in the Q&A box for specific speakers. So I'll start with bit.bio. Savitri, have you used your cells in 3D models, e.g. organoids? And what is your business model? Do you sell differentiated hepatocytes?
Thank you for the question. I'll start by answering your second question first. So our business model, we provide iPSC-derived cell types. We have different cell types in CNS area, and also we have skeletal muscles, we have hepatocyte. Yes, we currently only have iPSC differentiated hepatocytes in 2D, but we are also working towards the development of co-culture with hepatocytes and non-parenchymal cells, and we are also working towards the development of disease model. And to answer your second question, if we have used our cells on 3D model, this is something that we are currently exploring. And in fact, we are hoping to receive data anytime soon.
Great, thank
you. Thank you.
All right, we have one for CN Bio. So what kind of media was used in the PBMC liver tissue culture in LC12? And do myeloid cells adhere to the plate?
Good question. So we use a combination of primary human hepatocyte media and RPMI typically used to culture PBMCs. And in terms of adherence of myeloid cells, so myeloid cells typically do adhere to tissue culture plastic, and we do find this within the LC12, but we're able to remove them using trypsin at the end of an experiment for that full recovery.
Great, thank you. All right, and we have one for Anna. For the spheroid C2C calculation, do you compare directly to the Cmax or to a particular multiple of the Cmax?
Thank you for the question. Yeah, we compare directly to the Cmax, the human Cmax, and that is the total plasma Cmax. And then there is, as you can see in the paper, we have a threshold of 176-fold, where the safe compounds are above this threshold, and then you found the hepatotoxic compounds below this threshold.
Great, thank you. All right, well, if there's no other questions, I think we can go ahead and wrap up early. Thank you to the presenters for all of your great presentations, and thank you for the audience for sticking around, and I hope everyone has a great rest of your day. Thanks.
Thank
Bye.
you. Bye.